everyone and welcome to Miss Estruch Biology. In this video I'm going through every single topic that could come up on OCR A paper 1. So you know it's going to be a long one, but there's always the option of ×2 speed, and if you do need more support with this, then don't forget to check out my OCR A notes or my flashcards, both of which are going to help you to remember the key marking points and get you ready for those exams. So first of all, there are 3 different types of microscopes that you need to learn about, starting with the light microscope. And light microscopes have the lowest or poorest resolution, and that's because they have the longest wavelength of what creates the image, and that is light. Light has the longest wavelength compared to electrons. But the plus side is you can get living samples that you can examine, and you can get a colour image as well. The 2 electron microscopes are transmission and scanning. And for a transmission electron microscope, it has a high magnification and resolution. And to create these images, the electrons pass through the specimen to create it. The scanning electron microscope also has a high magnification and resolution, slightly lower than the transmission, but much higher than the light microscopes. And in these ones, the electrons bounce and scatter off the surface of the specimen to create a 3D image. The resolution is the minimum distance between 2 objects in which you can still view them as separate. And in a light microscope or optical microscope, this is determined by the wavelength of the light. Whereas electron microscopes, it's determined by the wavelength of an electron. Magnification refers to how many times larger the image is compared to the actual object that you are viewing. So with the light microscopes or optical microscopes, there are 4 types of slide preparation that you need to be familiar with. Dry mount, wet mount, squash slide and smear slide. The dry mounts are when thin slices or even the whole organism or specimen are viewed. And what you would do is place that specimen on top of your glass slide with just a cover slip placed on top. So this might be used if you're examining a really thin slice of plant tissue, or you might have tried this before with your own hair. Wet mounts are more common, and you would have done this potentially in lower school when you looked at maybe a swab of your cheek cells or onion cells. And this is when the specimens are added to water or maybe a stain before you have the cover slip lowered on top with a mounted needle to prevent air bubbles forming. And if you were going to examine living aquatic organisms, you would have to do that as a wet mount. Now in A-level, you might be more familiar with a squash slide. So these are wet mounts, but then you would push down on the cover slip to ensure that you have a really thin layer of cells so that the light can pass through. So when you did your root tip squash practical, you would have taken a very thin slice from the tip of the root of an onion or garlic, placed the stain on top, squashed it by pressing down, and that way you should get light passing through so you could visualize and see the chromosomes. And then finally, we have the smear slides. These are created by placing a drop of the sample at one end of the slide and then using the edge of another slide held at an angle to smear the sample across that initial slide. A cover slip is then placed on top. And this is used when examining blood cells in a blood sample, for example. Next, we move on to this math skill, the eyepiece graticule, and calibrating it. So inside a light microscope, in the eyepiece, there is actually a scale that you can insert on a glass disc, and that is what the eyepiece graticule is. And the point of it is that when you look through the microscope, you can use that scale to measure the size of the object you're looking at. But there are different lenses on your microscope which are going to be causing different magnifications. So you might have ×10, ×40, ×100. And at each magnification, the value of one of the divisions on your eyepiece graticule will be different. So you have to know how to calibrate it at every magnification to work out what one division on your eyepiece graticule is worth. So this is how you would do that. You would need to use something called a stage micrometer. And this is a glass slide which essentially has a ruler on it. And you would place that on the stage of your microscope, look through your eyepiece, and then move your stage micrometer so that it is aligned right next to your eyepiece graticule. So on this image, the circle's representing your field of view when you're looking down the microscope. The bottom scale is the scale on your stage micrometer, and that has been aligned against the eyepiece graticule, which is already within the eyepiece of the microscope. Once you've then got them aligned, step 2 is to count how many divisions on the eyepiece graticule fit into one of the divisions on the stage micrometer. In this case, we can see 2 divisions fit into one of the stage micrometers. Now, on a stage micrometer, each division is worth 10 micrometers. That's why that value there says 10. It's one division, but that one division is worth 10 micrometers. So we can then use that to work out at this magnification what is one division worth on the eyepiece graticule. So if we know that one of these divisions is worth 10 and 2 divisions fit into one of those, is 10 divided by 2. So we know that at this magnification, one of these divisions on the eyepiece graticule is worth 5 micrometers. So you can then measure your specimen to work out what is the actual size of your specimen. Something else that you can do in terms of calculations with the light microscopes, but even with electron microscopes as well, is the magnification calculation. So you need to know that to work out the magnification, it's the size of the image divided by the size of the real object. And you might have to rearrange that formula to work out one of the other components of this equation. And it's never usually that straightforward. Often you have to have changing of units involved as well. So we usually measure the size of an image in millimeters, and the size of the real object is usually in micrometers. So to convert your millimeters into micrometers, you would have to multiply by 1,000. So there's normally that additional step as well. So staining is the next thing you need to know about. And some cell components are really difficult to see under the microscope unless you add a stain to make it a much more obvious colour so it stands out. And differential staining is a technique which involves many chemical stains being used to stain different parts of the cells different colours. Again, just to make it visually more obvious what you're looking at. So crystal violet or methylene blue are 2 stains that are commonly used, and they are positively charged stains. And that means they'll be attracted to anything that is negative. So negatively charged components of the cell, and they will stain those parts of the cell. You could then also add nigrosin and Congo red, which are negatively charged, and therefore they actually can't enter the cells because the cytosol would repel it. And that would create a stained background, a different colour, and the unstained cells will then stand out. Observations of structures from under a microscope can be recorded in the form of scientific drawings, and there are set rules that you need to know for how you create a scientific drawing, because these are very different to your typical artistic shaded style drawings. So number 1, it should always be in a pencil. Title the diagram to indicate what the specimen is. You have to state what magnification was used for the microscope and that you have drawn the image at. You should be labelling the key features as well. So for a high power plan of this would be limited to just the nucleus, cytoplasm and plasma membrane. You also have to annotate cell components, cells and sections of tissues that are visible also, and stating what colours and shapes they are, so not necessarily their functions. So then point 6 is the bit where we're saying it's not like an artistic diagram, so you don't do sketches. So you should use solid lines that touch, but there are no overlaps between those lines. Also, there shouldn't be any colouring or shading in. And then lastly, the label lines should be horizontal, drawn with a pencil and ruler, and not have arrowheads on them. So essentially, the aim of a scientific drawing is to show the size, location and proportion, and annotating some of these structural details. You could also be asked to do the scientific skill of scientific drawings, and these are very different to artistic drawings. They have a complete set of rules, and it includes when you are looking at your specimen under the microscope, you would then try and draw it using, first of all, it has to be a pencil, a really sharp pencil so you can be accurate. You have to include a title of what you've drawn. You have to state the magnification, or you could include a scale. You should always annotate the cell components or the cells themselves or the section of tissue that is visible that you've drawn. It shouldn't be sketchy lines. It has to just be solid lines that don't overlap with no gaps, and you mustn't do any colouring or shading. So essentially the aim of these diagrams is to show the size, location, proportion. They are not artistic. So that's why we don't have any sketching, shading or colouring. It's all about the facts, the size, shape, position and labelling the structures. And although I've said this for microscopes, when you come on to do dissections, you could be asked to do scientific drawings of what you observe in your dissection as well. So then we move on to the electron microscopes. And a beam of electrons is used here to create the image. And electrons have a very short wavelength. And that is why electron microscopes have a higher resolution. And because they've got a high resolution, small organelles and internal structures can be visualised. The image is created using an electromagnet, so that magnet is used to focus the beam of electrons. Now you can't actually use living samples with electron microscopes, and that's because the air would absorb the beam of electrons. So your electron microscopes have to be used with a vacuum. So your specimen has to be in a vacuum so that the air doesn't absorb the electrons. The image is also in black and white, so you do have to add a stain to add any colour, or you can do that artificially afterwards. So transmission electron microscopes are one type of electron microscope. And for these ones, your specimen has to be very, very thin. You're using an extremely thin slice through your specimen. They're stained and then placed in a vacuum. An electron gun will then be used to release a beam of electrons. The electromagnet will focus that beam, and these transmit or pass through the specimen. Some parts of the specimen will absorb the electrons, and that will make them look darker in the image, and some parts won't and they look lighter. So you get a 2D image because the electrons are passing through. And these are really useful for being able to see the internal structures of cells. Scanning electron microscopes, in these ones, the specimen doesn't have to be thin because the electrons don't pass through the specimen. Instead, the electrons are beamed onto the surface, and the electrons scatter in different ways, so they reflect backwards in different ways depending on the contours of your specimen. And this is what produces a 3D image of the surface of the specimen. So the next bit of spec then is knowing the organelles you find in eukaryotic and prokaryotic cells. You need to know the structure and function. And here are the 13 organelles that you need to know for eukaryotic cells. And eukaryotic cells include animals, plants, and fungi. So here's a cross-section of an animal cell showing some of those structures. And here's a cross-section of the plant cell, so you can see the location and general shape of these organelles. But we're going to go through each of them in detail, starting with the nucleus. The key structures are that you have a nuclear envelope, which is this double membrane layer, and the holes that we can see within it are the nuclear pores, and that is what the mRNA can pass out of after transcription. There's also nuclear plasm, which is a granular jelly-like material in the middle. The chromosomes are found inside of the nucleus, and these are protein bound, so wrapped around histone proteins, and they are linear. Lastly, we have the nucleolus, which is this smaller sphere inside of the nucleus, which is the site of RNA production, and it makes ribosomes. So the function then is, first of all, it's the site of DNA replication and transcription. And transcription is the first stage of protein synthesis when mRNA is created. It also contains the DNA for each cell, and it's the site of ribosome synthesis. Flagella are not actually found on all eukaryotic cells. They're found on some, so for example, sperm cells. And it's this whip-like tail structure, and the function is for mobility. It is also sometimes as a sensory organelle for chemical stimuli. Cilia, again, are not on all cells, and these are hair-like projections that come out of the cell itself. And they might be stationary cilia, or they might be mobile. And mobile cilia move to help sweep substances along. And that could be in the trachea, for example. They'll be moving in this wave-like motion to help sweep the mucus up and out of your trachea to prevent lung infection. Stationary cilia are important in sensory organs, such as your nose. Centrioles, these are made up of microtubules, which we can see here in this image, the microtubule. and they occur in pairs to form a centrosome. So that's what we're looking at here. We've got them as a pair forming that centrosome. Now, these are involved in the spindle fibre formation, which is essential in organising the position of chromosomes in mitosis and meiosis. The cytoskeleton is a network of fibres found within the cytoplasm all over a cell, and it consists of microfilaments, microtubules, and some intermediate fibres as well. Now, the function of this is it provides mechanical strength to cells. It helps to maintain the shape and stability of a cell, and many organelles are actually bound to that cytoskeleton to hold it in a fixed place. Microfilaments are responsible for cell movement, and microtubules are responsible for creating scaffold-like structures. The intermediate fibres help to provide mechanical strength.
We then move on to looking at the endoplasmic reticulum. In terms of the structures, there's two types. We have rough and then we have smooth endoplasmic reticulum. Both of them have folded membranes, which are called cisternae, but the key difference is the rough have ribosomes attached to the outside, whereas the smooth doesn't. The difference in function then links to the fact that the rough has the ribosomes, because the rough endoplasmic reticulum has the function of protein synthesis for proteins destined to leave the cell, and the proteins are transported through the rough endoplasmic reticulum and into secretory vesicles. The smooth endoplasmic reticulum is where lipids and carbohydrates are synthesized and stored. Next then we look at the Golgi apparatus and the secretory vesicles. This is the Golgi apparatus and the secretory vesicles here, which I've seen many students describe as looking a bit like a Wi-Fi symbol, and that's how they recognise it in comparison to the smooth endoplasmic reticulum. But what it is folded membranes making the cisternae. You then have secretory vesicles that pinch off from that cisternae. The function then is, it's where we have the addition of carbohydrates to proteins to form glycoproteins. You also have the production of secretory enzymes. Carbohydrates are secreted. We have the transport, modification and store of lipids. We get lysosomes forming. Molecules are labelled with their destination. And then finally, the finished products are transported to the cell surface membrane, which we can see here, in those secretory vesicles where they fuse with the membrane and the contents are released.
Lysosomes. So we just said lysosomes can be created by these Golgi apparatus, and lysosomes are vesicles and they're bags or vesicles of digestive enzymes. And they can contain lots of different enzymes. Some functions include the hydrolysis of bacteria. So you have lysosomes fusing with phagosomes in phagocytosis. That releases the digestive enzyme to hydrolyze and destroy pathogens. They're also involved in completely breaking down dead cells. And then once they have hydrolyzed and digested whatever it is that they're breaking down, the lysosome will then fuse with the cell membrane, and that will release its contents to the outside of that cell. Mitochondria are double membrane bound organelles. So we have an outer membrane, and the inner membrane is shown here in yellow. It folds in to create the cristae. The mitochondria is the site of the fluid centre in the middle is called the mitochondrial matrix, which is the site of some of the stages of aerobic respiration. And they actually also contain their own ribosomes and loops of DNA so that they can create the enzymes necessary for respiration inside of the organelle itself. So it's the site of aerobic respiration specifically. Therefore, it's the site of ATP production. And as I've already said, it contains the DNA to code for the enzymes needed for respiration. So the ribosomes it contains are the 70S ribosomes, which are much smaller, which are also the type that are found in prokaryotic cells. So that leads us into ribosomes. Ribosomes are small. They're very, very small. You can see them like these tiny dots in the microscope images. They're made up of two subunits of protein and RNA. So those are the two molecules it contains. There are 80S ribosomes, which are the larger ribosomes found in the cytoplasm of eukaryotic cells, or 70S ribosomes, which are much smaller, found in prokaryotic cells, and also in mitochondria and chloroplasts for eukaryotes. And the function is, it's where protein synthesis occurs. Chloroplasts are found in plant cells, which are eukaryotic organisms. They also have a double membrane, and then inside you have these foldings of even more membrane, which are called the thylakoids. And these foldings stack up to look a bit like coins, and we call those stacks the grana for plural or granum for singular. And you also have this fluid, which is shown in this beigey orange colour, which contains lots of enzymes needed for photosynthesis. So this is the site of photosynthesis. Cell walls are found in plants and fungi of eukaryotic organisms, not in animal cells. And in plants, you have microfibrils of the cellulose polymer, and fungi, you have chitin instead. So they're made up of two different molecules. Plants have cellulose for structural strength, whereas fungi have chitin. And that is still a polysaccharide, but it also contains nitrogen. The function of the cell wall, though, is to provide structural strength. The plasma membrane is found in all cells, and this would form the cell surface membrane. It's made up of this phospholipid bilayer, and within that bilayer you have other molecules embedded, such as channel proteins, carrier proteins, proteins on the outside which might act as receptors, glycoproteins which might act as receptors. You have cholesterol which affects the fluidity and therefore the permeability of the membrane as well. And all of these structures together help to control what can enter and exit the cell. Or it could be the organelle, because you do have these membranes on the outside of some organelles as well. So some of those organelles are involved in the production of proteins. And you do get long answer questions where it's in this topic, but they want you to link together everything you've learned to say which organelles are involved in the production and secretion of proteins. So let's summarise that. Number one, the polypeptide chains are synthesized on the rough endoplasmic reticulum, or you could say the ribosomes, because those are found on the outside. Those polypeptide chains move to the cisternae in the RER, and they get packaged and they get folded and packaged into vesicles to be sent to the Golgi apparatus for further modification. Now they have to move through the cytoskeleton to get to that Golgi apparatus. Once they're inside the Golgi apparatus, the proteins are modified further and packaged into the vesicles, and those secretory vesicles carry the proteins to the cell surface membrane. The vesicle then fuses and releases the protein by exocytosis.
So looking at the prokaryotic cells then in comparison to the eukaryotic cells, here are the key differences. First of all, prokaryotic cells are much smaller in size. They don't contain any membrane-bound organelles. Both have ribosomes, but prokaryotic cells have the smaller 70S ribosomes, whereas eukaryotic cells have the larger 80S ribosomes. The DNA in a prokaryotic cell is not contained within the nucleus, and prokaryotic cells do have cell walls, but they're made up of peptidoglycan, whereas in eukaryotic cells, in plants it'd be cellulose, and in fungi it'd be chitin. Now, some prokaryotic cells have additional features, which are the plasmids, a capsule around the cell, and the flagella.
So let's have a look at some of those key differences. So we said there's no membrane-bound organelles. So that means they don't have their circular DNA in a nucleus. They don't have mitochondria, chloroplast, a Golgi apparatus, and endoplasmic reticulum. They're much more basic organisms. The ribosomes, we said they do still have ribosomes, but they're much smaller. So they are the 70S ribosomes compared to the 80S ribosomes found in eukaryotic cells. They don't have a nucleus. So instead of a nucleus, they have their single circular DNA just free within the cytoplasm, and it is not attached to proteins. And that is what this image here is showing. Now that is not the same thing as plasmids. Plasmids are additional loops of DNA that some bacteria have. They don't all have them, and they only have a few genes on them, and it's where you'd find the genes for antibiotic resistance. And bacteria either don't have them at all, or they might have them, but they have them in varying numbers.
The cell wall of a prokaryotic cell is made up of peptidoglycan, which is a glycoprotein. In comparison, eukaryotic cells, animals don't have a cell wall, but plants and fungi, which are eukaryotes, do. And plants have a cell wall that's made up of microfibrils of the cellulose polymer, and fungi have a cell wall made up of chitin, which is a nitrogen-containing polysaccharide.
The capsule, which again only some bacteria would have, this is a slimy layer made of protein on the very outside. And the function is to prevent the bacteria from drying out or desiccating. But it also helps to cover the antigens to make it harder for the host's immune system to detect the bacteria. The flagellum, which some bacteria have, and some might have multiple, some might only have one, some won't have any, just like a flagella in a eukaryotic cell, it rotates and its function is to move the bacterium. Part of this chapter is 2.12 biological molecules. Biological molecules all contain carbon, but below shows the elements that each molecule contains. So in addition to carbon, carbohydrates have hydrogen and oxygen, as do lipids, but just in a different proportion. Proteins also have nitrogen and sometimes sulfur, and nucleic acids also have nitrogen and a phosphorus. Ions also play a really important role, and here is a long list of the ones that you need to know.
2025 update here. I have actually just edited some of the uses, so just check through those carefully to be mark scheme specific. So if you're going to be making notes or flashcards, definitely just double check that you're using the information from this slide.
So the cations, which are the positive ions, and then the anions, which are the negative ions. The best thing to do to learn this would be to turn all of these into a flashcard. So have cation, calcium ions on one side, and then have the information on the other side to show what the function of that ion is, or what important role it plays. So for this bit, I definitely recommend you pause, turn these into flashcards, and then this will be the best way to get your head around all of this content. So then let's take a look at water as a biological molecule. It's a polar molecule due to the uneven distribution of charge. And we can see here we've got delta negative on the oxygen and the delta positive on the hydrogen atoms, which means a slight negative and a slight positive charge. Now, because of that uneven distribution, that enables the formation of hydrogen bonds between the oxygen and a hydrogen atom between two different water molecules. And that's what we're seeing here, a hydrogen bond forming between the hydrogen and the oxygen of different water molecules. And a hydrogen bond is actually pretty weak in terms of bonds, but collectively they do provide quite a lot of strength. And it's that structure and those hydrogen bonds which form all the different properties of water that we're going to have a look at. So these are the four that you need to know: as an important solvent in reactions, a transport medium, a coolant, and for providing habitats. So let's take a look at the details for each one, starting with water as a solvent. Because water is a polar molecule, that means that it can also interact with other polar molecules. And the reason for this is the slight positive charge on the hydrogen atoms will attract any negative solutes that have dissolved, and the slight negative charge on the oxygen atoms of water will attract any positive ions in the solutes. So if we think about, for example, sodium chloride, the chloride would be negative, the sodium would be positive, and that is why it dissolves so readily in water. Non-polar or hydrophobic molecules cannot dissolve in water and instead are actually repelled, and that's things like lipids. The cytosol in eukaryotic and prokaryotic cells is mainly water, so this ensures many solutes can dissolve within the cell and then be easily transported, which is why this is such an important property. This leads us into the importance of water as a transport medium. Once those solutes have dissolved, they can then be readily transported around plants or animals. And if it's in animals, it'd be within the blood, and if it's in plants, it could be in the xylem or the phloem. Now, for this bit of the topic, it would mainly be the xylem that it's referring to, because that would be all of those ions that we saw earlier on. But glucose, sucrose, those sugars do also dissolve, and they're transported in the phloem. Now, it is possible to transport these dissolved solutes in the xylem due to the cohesion that we saw in the water molecules. So we said that hydrogen bonds form between the hydrogen and oxygen atoms in different water molecules. And due to that, the water molecules stick together and they form a continuous column of water. This is an advantage because as water evaporates out of the stomata in the leaves, that leaves a negative space, a negative pressure. And that negative pressure pulls on the continuous column of water, and it's easily transported up the stem because it is all stuck or cohesed together. The next one is water as a coolant, and this is due to two different properties of water. First of all, water has a high specific heat capacity, and this means it takes a lot of energy to increase the temperature of water. You do learn that in more detail in chemistry, but that's sufficient for biology. The reason it requires so much energy is because energy is needed to break the hydrogen bonds between the water molecules to increase the temperature. Now that's an advantage because it means internal temperatures of plants and animals remain relatively constant. So even if there is a fluctuation in the air temperature, that should mean that the temperature inside of an organism and in the cells remains relatively constant. And that's good because then enzymes are not going to denature, or if it gets really cold, they're not going to reduce in activity too far. So essentially water buffers temperature changes. Now the other property is the fact that water has a large latent heat of vaporization. And this means a lot of energy is required to convert water in its liquid state to a gaseous state, water vapor. And again, that's due to the hydrogen bonds. Energy is required to break the hydrogen bonds between water molecules to turn it into a gas. And that provides a cooling effect when animals sweat, for example, or even when plants transpire. Water also provides a habitat, and we said previously that it acts as a buffer for temperature changes, and that is useful for any aquatic organism as well, because living in a body of water, if the temperature is buffered, then it should mean that your enzymes aren't going to denature if there are any significant fluctuations in temperature. The other property links back to the cohesion provided by the hydrogen bonds. Those hydrogen bonds not only create a continuous column of water in a xylem, but they also create a surface tension to the top layer of water molecules. And that enables small invertebrates to be able to move and even live on the surface. And that provides them with a habitat away from the predators within the water. Finally, ice is actually less dense than liquid water due to the hydrogen bonds. And as a result, ice floats on top of bodies of water, and that can provide a surface habitat for animals such as polar bears.
And it also insulates the water below, keeping it a liquid for aquatic organisms.
So now we're moving on to the next bit in this topic, which is looking at monomers and polymers. And mono means one, poly means many. So our literal definition is monomers are smaller units which can bind together to create larger molecules, or in fact polymers. And a polymer is made up of lots of monomers bonded together. Here are our examples of monomers and polymers that you need to know in this topic. Glucose is a monomer which forms the polymer starch, cellulose and glycogen. Amino acids is the monomer that forms the polymer protein. And nucleotides form the DNA and RNA polymers.
2025 edit, just to make you aware, before you see me go through this whole flow diagram, you actually only need to know glucose and ribose on your spec. So although I'm going to talk about others, glucose and ribose are the two monomers or monosaccharides that you need to know for OCR 2025.
So if we have a look at the carbohydrates first, this is showing you an overview of all the carbohydrates that you need to know about. We already said that carbohydrates contain carbon, hydrogen and oxygen. And we can split them into monosaccharides, disaccharides and polysaccharides. Mono meaning one, saccharide is sugar, so it's just one sugar unit. And that would be your glucose, fructose and galactose. And glucose is the main one that you learn about. Disaccharides, di meaning two, is two sugars bonded together. And you'll be learning about sucrose, maltose and lactose. Finally, polysaccharides means many sugars. And what you actually have is at least 3 sugars bonded together. In reality, it's going to be far more than 3 sugar units. And you'll be learning about starch, cellulose and glycogen.
So here's alpha glucose, one of the isomers, and it's C6H12O6. So it contains 6 carbon atoms, 12 hydrogen atoms and 6 oxygens. And here is your structure. And this is the level of detail that you need to be able to draw it in. So the way that I always remember it is start off by having your hexagon and you have an oxygen in the top right. Every other bend or angle in that hexagon is a carbon. Coming off carbon 1, you have hydrogen on top, hydroxyl, same for carbon 2, same for carbon 4, but it's carbon 3 which is the opposite way round. So the hydroxyl group's on top, hydrogen is on the bottom. Then we get to carbon 5 and 6, so you only have a hydrogen coming off carbon 5, and then carbon 6 is branched off the top, C6. You then would have a hydrogen here, hydrogen there, and a hydroxyl there, or in other words, CH2OH. So you do need to know how to draw alpha glucose in this level of detail.
If we then have a look at our beta-glucose, which is our other isomer, we can see that for beta-glucose, which is over on this side, is exactly the same with the exception of this, the hydroxyl and the hydrogen on carbon 1, which is this first carbon in the ring, are the other way round. The hydroxyl's on top this time. Now, that slight difference has a big impact on the position that bonds can form and therefore the overall shape, structure and function of the polysaccharides.
Then we have the structure of ribose, one of the other monosaccharides you need to know. And monosaccharides can be categorized according to how many carbon atoms they contain. Now, ribose is classed as a pentose sugar because it contains 5 carbons. And we can see where we've got these numbers, that's indicating where the carbons are in the structure. So we've got carbon 1, 2, 3, 4 and 5. Glucose has 6 carbon atoms that would be known as a hexose sugar. So where you see OSC on the end, that means it's a sugar, and then the prefix in front is to indicate how many carbons it contains. And here is our structure of ribose, the pentose sugar, which is a monosaccharide that you need to know the details of.
So next we move on to the disaccharides. Di meaning 2, and we said it's made up of 2 sugar units or 2 monosaccharides. They're joined together by a glycosidic bond, which is formed during a condensation reaction. And a condensation reaction is when a water molecule is removed, a chemical bond is formed to create a larger molecule. And that's what we can see here. Glucose plus glucose forms a larger molecule, the disaccharide maltose, and water is produced. The other 2 disaccharides you need to know about are lactose and sucrose. All 3 of the disaccharides are made up of 1 monomer of glucose, and then it's the second one which is different for all 3. Maltose is made up of an extra glucose, lactose is made up of galactose, easier to remember because lactose is in the name, and then sucrose, the second monosaccharide it's made up of, is fructose.
The last extra bit of information that you do need to know about disaccharides is for the glucose molecule that is used to make each of these disaccharides, you need to know the isomer. So for maltose, it's 2 lots of alpha glucose. Lactose is beta glucose and also galactose. And sucrose is alpha glucose plus fructose.
So we said that disaccharides are made in a condensation reaction. That is a reaction we're going to see over and over in the biological molecules topic when making polymers and also when making other molecules. So it's a key definition to learn, definitely one to put on a flashcard. So a condensation reaction is joining 2 molecules together by removing water, and a chemical bond is formed. Hydrolysis reaction is the splitting apart of water molecules through the addition of water, and a chemical bond is broken.
Just an extra detail to make this really marked scheme specific with 2025 is you need to say that it is a water molecule for the condensation reaction and a water molecule for hydrolysis, not just the addition of water or the removal of water. It is 1 water molecule per reaction.
Let's have a look at a condensation reaction in action. We've got a generic monosaccharide and another generic one over here. We aren't really shown any of the extra details, only where the bond is going to form because that's the part of interest. So we can see here the hydroxyl group, that is where the water is going to be eliminated from. And in removing that water molecule, a bond forms between the carbon, oxygen and carbon. And that in this case is a glycosidic bond. And we name the glycosidic bond by the position it is found. So a 1 to 4 glycosidic bond means it's a bond found between carbon 1 and carbon 4. And the way we number them is always starting from the first carbon after the oxygen in the ring. So this would be carbon 1, 2, 3, 4, 5 and 6. And same on this one, 1, 2, 3, 4, 5 and 6. So this is a 1,4 glycosidic bond. The opposite of that, hydrolyzing, would be breaking that bond through the addition of water. And in doing that, the water is added back in to those hydroxyl groups, and now we go back to having those 2 monosaccharides. Next up we move on to our polysaccharide, and this is created by many condensation reactions between many glucose monomers. So just as the basics to start, starch is found in plants and it's a store of glucose. Cellulose is also found in plants, but the function is different. It's for structural strength. Glycogen is found in animals, and this one is a store of glucose also. So this summary table goes through the structure and function of all three of those carbohydrates. Again, this would be a great slide to pause and turn into multiple flashcards. Each of these individual boxes could be a flashcard. So let's have a look at starch first. The monomer is alpha glucose. That is the isomer of glucose that this molecule is made up of. And because it's got alpha glucose, it's able to form both 1-4 and 1-6 glycosidic bonds. Amylose is one of the polymers in starch, and that only has 1-4 glycosidic bonds. And due to that, amylose forms these long, straight chains that then coil up to make a helix. So overall, it's got a helix shape, whereas amylopectin is made up of 1-4 and 1-6 glycosidic bonds. And as soon as there's a 1-6 glycosidic bond, that results in a branch coming off. So amylopectin is a branched molecule. These two we'd already said on the slide before. So I'm going to skip on now to how the structure links to the function. So we said it's a store of glucose. The fact that the amylose forms a helix means you can compact the molecule to fit a large amount of glucose in a small space. The fact that amylopectin is branched increases the surface area for enzymes to attach onto the end glucose molecules, hydrolyze them off, breaking the bond, and that glucose can then be used in respiration. The final one is actually the same for all three. Because it is so large, it's insoluble in water. And that means it can be stored within a cell and not affect osmosis. So it's not going to cause the cell to swell and burst. Cellulose is the next one. This is also found in plants, but the difference here is it is for structural strength. It has a very different structure, and that is all down to the fact that the monomer is beta glucose. Because it's beta glucose, we get 1-4 glycosidic bonds only. And that results in long, straight chains of this beta-glucose molecule. Those long, straight chains are held in parallel to each other, and many hydrogen bonds form between all of those chains. One hydrogen bond is weak, but because there are so many, it provides this collective strength. And those then form macrofibrils, which combine to form a cellulose fibre. So the function then linking to that structure is the fact that there are so many hydrogen bonds provides that collective strength, and that is why cellulose within the cell wall helps to give structure and prevent the cell from bursting. Lastly, our glycogen is also made of alpha glucose, and it's very similar in structure to amylopectin in starch. It's composed of 1-4 and 1-6 glycosidic bonds, but it actually has far more 1-6 glycosidic bonds than amylopectin. So that means it's even more highly branched. Now this is important because the fact that there's even more branches means there's an even greater surface area for the rapid hydrolysis back into glucose so the glucose can be used in respiration. And because this is the store of glucose in animals, mainly in muscle and liver cells, if that glucose can be rapidly released, if an animal does need to run to either protect itself or to hunt, then it will have that glucose. Our next biological molecule are the lipids. So these are macromolecules, but they're not polymers. They're non-polar molecules, so they don't have a charge. For that reason, they're insoluble in water, but they do dissolve in organic solvents such as ethanol. And they are hydrophobic, meaning they are repelled by water. Lipids are made up of two molecules, fatty acids and glycerol, and they do not form polymers. The two key lipids that you need to know about are the triglycerides, shown here on the left, and phospholipids, shown on the right. They are very similar in structure. They both have a glycerol molecule here, which is sometimes described as the glycerol backbone, and they then have fatty acids attached to them. A triglyceride has 3 fatty acids, whereas a phospholipid has 2 fatty acids, and instead of the third, it has a phosphate group attached to it. So the way that those molecules are made is very similar, but this one is just focusing on the triglycerides. That glycerol molecule binds to the 3 fatty acid chains through a condensation reaction. So water is eliminated, a bond is formed, and we've made a larger molecule. Bond that forms is an ester bond. So let's have a look at how that occurs. Here's the glycerol molecule, and then we have 3 fatty acids. Condensation reaction is the removal of water. So we need to have a look at where that water comes from. So it's coming from the hydroxyl group, from the glycerol, and from the fatty acid. When that is removed, we then have a triglyceride, plus 3 water molecules that were removed, and here is the ester bond. And we have 3 ester bonds. We have 1 for each fatty acid that's been bonded on. Fatty acids can be either saturated or unsaturated. A saturated fatty acid is when the hydrocarbon chain has only single bonds between the carbon atoms. Unsaturated fatty acid is when the hydrocarbon chain has at least 1 double bond between the carbon atoms. The properties of triglycerides do link to the structure. So first of all, the function is that they can transfer energy. And this is due to the large ratio of energy storing carbon to hydrogen bonds compared to the number of carbon atoms. So a lot of energy can be transferred if that was to be broken down. Due to that high ratio of hydrogen to oxygen atoms, they can also act as a metabolic water source. And this is because triglycerides can release water if they are oxidized, and this is essential in animals such as camels that live in the desert where there's very little water. As lipids are large, hydrophobic molecules, they are also insoluble in water, and that means they're not going to affect osmosis. They're also relatively low in mass, so a lot can be stored in an animal without it increasing the mass as much as muscle would, and therefore making them so heavy it could impact their movement. So phospholipids are made up of 1 glycerol molecule, 2 fatty acids, and a phosphate group. The way they're formed is very similar. It'll just be 2 condensation reactions instead of 3, but we still get ester bonds forming, but it'll be 2 instead of 3 that we saw in the triglycerides. Now, because of this phosphate group, it results in very different properties. That phosphate group has a charge, it's a negative charge, and that means that the head, which is what we call this structure at the top, is hydrophilic. And if it's hydrophilic and it's got a charge, it can interact and attract water. The fatty acid tails are non-polar, so they don't have a charge, and for that reason they're described as hydrophobic, and they repel water but mix with fats. Now, because of these 2 different charged regions, they are able to form the phospholipid bilayer. When you add phospholipids to water, because the heads are attracted to water, they will spin to be exposed on the outside. The tails are repelled by water, so they'll spin to be on the inside where they can interact with the other fatty acids, but they're not exposed to the water. And that's how we get the phospholipid bilayer that makes up cell surface membranes, but also some organelles membranes. Cholesterol is our next biological molecule, and it is very different in structure because it is a sterol. Sterols have 4 carbon rings and a hydroxyl group at one end, and they have both hydrophobic and hydrophilic regions. Now, these are embedded in the cell membranes to impact the fluidity. They help reduce the fluidity of membranes at high temperatures, and they increase fluidity at low temperatures. And in that way, they help to control the movement across a cell membrane. Proteins are our next biological molecule, and they are large polymers, and they are described as macromolecules. They're made up of the amino acid monomers, and here is our general structure of an amino acid. We have a central carbon with an amine group, a carboxyl group, a hydrogen, and then the R group is the variable group. And that means it is the part that changes in all 20 different amino acids. Proteins are organized into 4 different levels. They are first made by the ribosome and creates this polypeptide chain. And then we're going to look at how that gets folded, processed with different bonds holding the structures together at each of these levels of organization. So the primary structure is the order or the sequence of amino acids in a polypeptide chain. And you do have to emphasize sequence or order to get that mark. The secondary structure is the further folding of the primary structure. And you can either get it folding into an alpha helix or beta pleated sheets. And these shapes are held in place by hydrogen bonds. We can see those just here. The hydrogen bonds form between the oxygen in a carboxyl group and the hydrogen on an amine group, and that is what we can see down here. So it's between different amino acids, between those two atoms, and that holds that beta-pleated sheet and alpha helix in place. The next level of organisation is the tertiary structure. And this structure is the further folding to form a unique 3D shape held in place by 4 different types of bonds. There are the hydrophobic and hydrophilic interactions, and these are very weak interactions. The hydrogen bonds, which we already saw, and again, those are quite weak. Ionic bonds are stronger bonds, and those form between the R groups of the different amino acids, and the disulfide bonds, which are only sometimes present because there has to be a sulfur in the R group between 2 amino acids for this to form. So the ionic and the disulfide bonds form between the R groups of different amino acids, whereas the disulfide bond, as we said, only sometimes occurs if there is an R group that contains sulfur between 2 amino acids. The last level of organisation is the quaternary structure, and this is when a protein is made up of more than one polypeptide chain, which is shown here by the different colours. So we've got 4 polypeptide chains collected together to form one protein. And the key example that you learn is haemoglobin, which is made up of 4 polypeptide chains. Now, haemoglobin also has a prosthetic group attached to each polypeptide chain, and that is the heme group. And a prosthetic group is one that is not made up of amino acids, and for a haemoglobin molecule, the prosthetic group doesn't contain amino acids, but instead it contains the ion. A protein that has a prosthetic group, such as haemoglobin, can be described as a conjugated protein, which simply means a non-protein group has been added to it. You can also group your proteins as either fibrous or globular based on their final 3D shape in the tertiary structure. A fibrous protein has polypeptide chains that form long twisted strands that link together. They're very stable structures, insoluble in water, and all of those 3 points collectively help to give structural properties. So fibrous proteins provide structural strength. For example, collagen, which is found in your bones, and keratin, which is in your hair. Globular proteins, on the other hand, are normally spherical in shape. They're relatively unstable, they are soluble, and involved in metabolic functions. So, for example, enzymes and antibodies and some hormones. And if you think about enzymes, we do say they're sensitive to certain conditions, such as changes in pH and temperature, and that's what we mean by relatively unstable.
For the 2025 specs change, I've just added in some extra details here, linked to the level of detail you need to know for fibrous proteins and globular proteins. So if you are making notes or flashcards from this video, use this slide for the level of detail that is required. So 3 key examples of fibrous proteins that you need to know are collagen, keratin, and elastin. Collagen forms part of the skin, the tendons, cartilage, ligaments, bone, and connective tissues in the bronchi, bronchioles, and trachea. It's a quaternary structure protein because it contains more than 1 polypeptide chain, and it contains 3 polypeptide chains. And these are wound around each other like rope. The chains are held by hydrogen and covalent crosslinks between those molecules, and the crosslinks are staggered for strength, and chains lie close to each other due to 35% of its amino acids being glycine, which is the smallest amino acid. It's a very flexible molecule, but it's not stretchy. Keratin is used to form hair, skin, and nails, which all protect the body. It's important that it's insoluble, so these structures are not broken down by water in the environment. Elastin is our final example, and it's a common fibrous protein and makes up the elastic fibres around the alveoli and blood vessels, for example in the walls of the arteries, arterioles, venules, and veins. It also allows these structures to stretch and recoil to their original shape and size. The 3 globular proteins you need to know about are hemoglobin, pepsin, and insulin. Hemoglobin has been described previously, so it's a quaternary structure protein because it's got more than 1 polypeptide chain, and in fact it has 4. It has 2 alpha chains and 2 beta chains, and each one has a heme group attached to it, which is a prosthetic group, which is where the oxygen binds. Enzymes are also globular proteins. Pepsin is an example of an enzyme found in the stomach. It is a protease enzyme which can digest proteins using its specific shaped active site, which is complementary in shape to its substrate. Lastly then insulin, which is also a globular protein and the hormone produced by the beta cells in the pancreas to lower blood glucose concentration. Its specific 3D shape is complementary to the receptors on the cell surface membrane of the target cells, which for insulin are liver and muscle cells. Next, we're moving on to the biochemical tests for those biological molecules, starting with the test for starch. To test for starch, you would add iodine solution, which is this orangey brown colour, and if starch is present, then it would go bluey black colour. To test for a reducing sugar, you need to add Benedict's solution and heat for 5 minutes at 80 degrees C, and you do need to know the duration and the temperature. A positive test observation would be that solution goes from this blue colour to either yellow, green, orange or brick red. And the more red the solution is, the higher the concentration. You can actually also use reagent test strips, and this can be used to test for the presence and concentration as well of reducing sugars. For non-reducing sugars, you would do this following a negative Benedict's test result, which basically means the Benedict's solution remains blue. You would add hydrochloric acid and boil, and this is acid hydrolysis. You'd be splitting apart or hydrolyzing any disaccharides into the monosaccharides. You would then cool the solution and add an alkali, such as sodium hydroxide, to neutralize. You would then add Benedict's solution and heat for 5 minutes at 80 degrees C. If it was a positive test observation, the solution turns from blue to green, yellow, orange or brick red. The more red, the higher the concentration. You usually get at least orange or brick red because if you did have a non-reducing sugar, such as sucrose, you originally had just one sugar, sucrose. But once you've hydrolyzed it, you've split it into 2 sugars. So you now have double the quantity of sugar present, which is why it should go a more red colour. Ignore the fact that it says 1 next to all of these. It should be 1, 2, 3, 4, 5. To test for proteins, you would add biuret solution and make sure you are spelling that correctly. Biuret, not buret, like a piece of apparatus used in chemistry titrations. And then the positive test result would be it goes from a blue solution to that purple colour solution. To test for lipids, you would use the emulsion test. And to do this, you would dissolve your sample in ethanol first of all. Then you'd pour the sample on top of distilled water. And then finally, a positive test observation would be a white emulsion forming. Now you can also use colorimeters to get quantitative data instead of qualitative. So if you were using a colorimeter, first of all, you'd need to set the filter in the colorimeter. So set it to a particular wavelength of light. Then you'd calibrate to 0 using distilled water to basically show that this is the amount of light that's absorbed when there is nothing in there that is providing a colour. So you're calibrating it to show what 0 is. Then you'd insert your samples from your biochemical test. So for example, different concentrations of glucose with Benedict's solution and filter to then remove the precipitate, and what is left is what you would put in. You'd measure the percentage transmission of light and then finally draw a calibration curve using the results from known concentrations of glucose. You can also use biosensors, and this is when you'd have a single strand of DNA or protein which is complementary to the test sample and that is immobilized. When the sample is added, it will bind to the immobilized DNA or protein. This binding causes a change in a transducer and as a result an electrical current is released. And this current is processed to determine the concentration of sample present. Chromatography can also be used, and these practical investigations are looking at the separation of proteins, carbohydrates, vitamins or nucleic acids conducted using thin layer chromatography or TLC or paper chromatography. In chromatography, there's a stationary phase, so for example, the silica-covered plate or paper that does not move, and there's a mobile phase, and this is the solvent which does move. A concentrated sample of the biological molecule is placed 1 centimetre from the end of the stationary phase. The stationary phase is then placed in a beaker with less than 1 centimetre depth of the mobile phase, which is the solvent. As the solvent moves up the stationary phase, it has an affinity for the biological molecules and dissolves them, and this carries them up the stationary phase. Molecules that are the most soluble in that solvent will be carried the furthest, and the molecules that are the least soluble in that solvent won't be carried as far. Once the solvent is carrying no more of the molecule, the plate or the paper is removed from the solvent and the distance travelled by the molecules is measured. So to identify which biomolecules you have present on your chromatography paper, you can work this out by calculating the retention factor, or RF value. And here's the formula for this, which you need to know. It's the distance moved by the solute divided by the distance moved by the solvent. And by comparing the calculated RF values with RF values of known molecules in that same particular solvent, the biological molecules that you have present can be identified. So this is a technique that can also be used to test for biological molecules, but also for drugs and contaminants in food.
Next, move on to nucleotides and nucleic acids. So first of all, nucleotides are the monomers from which nucleic acids such as DNA and RNA are formed. And those nucleic acids contain nitrogenous bases, which can be categorized according to their structure, how many rings they have. So a purine has 2 carbon rings, and that would be adenine and guanine. As we can see here in the diagram, we've got the first ring and then the second ring. Whereas pyrimidines, those are nitrogenous bases which only contain 1 ring within their structure. And that would be cytosine and thymine in DNA, and uracil for RNA. The rest of the nucleotide is made up of pentose sugar, which is ribose for RNA and deoxyribose for DNA. And then finally, both would have a phosphate group. So the thymine or uracil in RNA is complementary to the base adenine, whereas guanine is complementary to the base cytosine. And what that means is you will always have a purine and a pyrimidine opposite each other in the double strand for DNA, and that ensures that the 2 strands are always equal width apart. Both DNA and RNA nucleotides undergo condensation reactions to go from having the nucleotide to the polymer chain. And it's a phosphodiester bond that forms between the adjacent nucleotides to create that polymer. The polymer of these nucleotides is called a polynucleotide, and that phosphodiester bond is a really strong covalent bond, and that forms between the pentose sugar, which would be deoxyribose in DNA or ribose in RNA, and the phosphate of a different nucleotide. So it'd form between the phosphate group and the pentose sugar. Next we have a look at ATP, and this is very similar in structure to a nucleotide. It contains a pentose sugar, which is always ribose, and it contains a nitrogenous base, which is always adenine. Adenine and ribose together make adenosine, and that's why ATP stands for adenosine triphosphate, because we also have 3 phosphate groups attached. And those 3 phosphate ions or groups play a significant role in energy transfer, and that's why ATP is essential for metabolism. It's an immediate energy source, providing energy for a whole range of different reactions. Now, ATP is made during respiration, both aerobic and anaerobic, but most of it is made in aerobic respiration. And this is through a condensation reaction using the enzyme ATP synthase. So here we see ADP plus an inorganic phosphate, that's what PI stands for, and that forms ATP, plus a water molecule is released because it's a condensation reaction. This is a reversible reaction because ATP can be hydrolyzed using the enzyme ATP hydrolase, and in that case the enzyme plus the addition of water would split one of the bonds between the phosphate groups. That releases a small amount of energy, and therefore we get ADP plus PI. Now, as well as releasing a small amount of energy, the inorganic phosphate group that's been released can then be bonded onto a different compound, and in doing that it makes the compound it's bonded to more reactive, and we call that phosphorylation. And this is actually an example of phosphorylation. ADP is phosphorylated to form ATP, and that makes ATP more reactive. It has more energy. So going back to DNA, deoxyribonucleic acid codes for the sequence of amino acids in the primary structure of a protein. And it's the primary structure of a protein which determines the final 3D shape and structure of a protein. polymer, the DNA polymer, forms a double helix made of 2 antiparallel strands, and those 2 strands are joined together by hydrogen bonds that form between the complementary bases. So if we have a look at how the DNA structure relates to its function, it's a very stable structure, and that is because of the phosphodiester bonds that form between the adjacent nucleotides, creating what we call the sugar phosphate backbone. And those covalent bonds mean that the whole structure is very stable. The fact that it's double-stranded is advantageous when it comes to DNA replication. It means that both of those strands can be used as a template. The fact that there are weak hydrogen bonds between the complementary bases is an advantage for DNA replication as well, because it means that very little energy is required to break those bonds, separate the 2 strands, and therefore have those 2 strands acting as a template. It's also a large molecule, and that means it can carry a lot of information. Finally, the fact that there is those complementary base pairing between adenine and thymine and guanine and cytosine means that identical copies can be created when DNA is replicated. Now, DNA can be precipitated out of cells to be examined, and that's one of the methods that you need to be aware of. So you can extract it from plant material using this method. First of all, you'd have to homogenize the cell. So you'd have to break it open with a blender and also a detergent. That will break open the cells, and cell membranes will also be broken open by the detergent, and that releases the contents of the cells. You would then filter to remove any large debris, add salt to break hydrogen bonds between the DNA and water molecules, add protease to digest the proteins, those histone proteins associated with the DNA, and then add ice cold ethanol to precipitate out the DNA from the solution. And the DNA appears as white strands, like we can see just here in the image. So next we move on to more details on RNA and the polymers of RNA. There are 3 types, M, T, and R. And if we start with R, the R stands for ribosomal, and that ribosomal RNA is what ribosomes are made up of. It's the main bulk of a ribosome. The ribosomes are also made up of protein, so those are the 2 components, rRNA and proteins. mRNA is a copy of a gene from DNA. It's created in the nucleus, and then it leaves via the nuclear pore to carry a copy of the genetic code of one gene to a ribosome in the cytoplasm. Now, it's much shorter than DNA, and the reason for that is it's only a copy of one gene. Human DNA, for example, consists of approximately 23,000 genes, whereas mRNA is only a copy of one of those genes. So it's going to be much shorter, and therefore it's small enough to come out of the pores in the nuclear envelope. It is short-lived though, and that's because it is leaving the nucleus and entering the cytoplasm. And in the cytoplasm, there are enzymes that can hydrolyze the polymer. And that is why we do not want the DNA leaving the nucleus, because if it did, your hard copy of a genetic code is at risk of being hydrolyzed and broken down. Whereas mRNA, it's short-lived, it just needs to survive long enough to be involved in protein synthesis, then it's digested by the enzyme, and those nucleotides can be recycled to make another mRNA strand. So the mRNA is a copy of one gene of DNA, but it's a single strand, and every 3 bases in the sequence codes for one specific amino acid. And these 3 bases are known as a codon. So a codon is 3 bases on mRNA that codes for a specific amino acid. tRNA is found in the cytoplasm. It's also a single-stranded molecule, but it gets folded to create what we call a cloverleaf shape. And it's held in this cloverleaf shape by hydrogen bonds. Its job is to transfer or bring specific amino acids to the ribosome. And that's what we have here, an amino acid attached at the top to this binding site. Now it's specific because it is determined by the 3 bases on the bottom of the tRNA called an anticodon. And these 3 bases are complementary to a particular codon on the mRNA sequence. Next we move on to semi-conservative DNA replication. DNA replication is described as semi-conservative because in replication, one entire strand of DNA is conserved and one entire new strand is created from new nucleotides. Copying errors in DNA replication can occur, but they occur randomly, spontaneously, and they result in a change to the DNA base sequence, and that's what a mutation is. That would be an example of a gene mutation. DNA replication occurs in S phase within interphase of the cell cycle, and when describing the DNA double helix, the top and the bottom of each strand have a particular term. We call it the 3', and this symbol here means prime, so 3' end or the 5' end. And this is determined by which carbon within the deoxyribose sugar of the nucleotide is closest to the top or the bottom. So this side, we're told it is the 3' end. If we zoom in to see that in more detail, what we mean by that is the number carbon that's most exposed. We've got carbon 1, carbon 2, carbon 3, carbon 4, and carbon 5 up here. So the bottom of this chain, carbon 3, would be described as being the most exposed. So this would be the 3' end of this chain. Whereas this chain, it's this carbon most exposed at the end, which is our carbon 5. So that's what this 3', 5' refers to. And it's a way to describe the top and the bottom, but take into account the chains are anti-parallel, so they do look slightly different. The enzyme that catalyzes DNA replication is complementary in shape to the 3' end. And that is the relevance of this to semi-conservative DNA replication. That enzyme can only bind at the 3' end of a chain, and then it will move along towards the 5' end. So the key stages in DNA replication are, first of all, the enzyme DNA helicase breaks the hydrogen bonds between the complementary bases of the two DNA polymer chains, and that causes the two chains to separate apart. Both of those strands then act as a template for the DNA replication, and free-floating DNA nucleotides will align opposite their complementary bases on both of those template strands. And within the nucleus, there are free-floating DNA nucleotides. That's where they come from. Hydrogen bonds will then form between the complementary bases of this new strand and the old strand. And then DNA polymerase is the enzyme that will attach to join together adjacent DNA nucleotides. And that means it is forming the phosphodiester bond between the nucleotides to create the new polymer chain. So properties of the genetic code then. There are three special features. It's degenerate, it's universal, and it's non-overlapping. Degenerate means that amino acids are coded for by more than one triplet of bases on DNA. Universal means the same triplet of bases codes for the same amino acid in all organisms. And non-overlapping means each base in a gene is only part of one triplet of bases that codes for one amino acid. So each codon or triplet of bases is read as a discrete unit. Now the reason these different properties are advantageous are, first of all, the fact that the genetic code is degenerate means even if a gene mutation occurs, changing one of the bases in a triplet, it might mean it still codes for the same amino acid, and therefore the mutation would have no impact on the final sequence of amino acids in the polypeptide chain, and therefore the protein shape and function. Universal is advantageous in genetic engineering because it means that we are able to remove a human gene, for example the human gene for insulin, and insert it into the plasmid of a bacterium, and therefore the bacterium will make human insulin. Non-overlapping is also advantageous, linking to the concept of mutations. The fact that each base is only part of one codon means that if there was a mutation in that codon, which meant it now coded for a different amino acid, the mutation will only affect one codon. So in your whole sequence of amino acids that get coded for, only one would be incorrect. And that should reduce the overall impact that the mutation has. And that leads us into this concept of protein synthesis. Proteins are created on the ribosomes of the rough endoplasmic reticulum in two stages. Transcription happens first, and this is when mRNA is created from a copy of one gene on DNA. And then translation is the second stage, and that's where the mRNA has left the nucleus, it attaches to a ribosome, and it is used then to create a polypeptide chain. And that's what we're going to have a look at, these two processes in detail. There are a couple of key terms though that you need to be familiar with to fully understand this topic. And the first is introns and exons. Introns are the sequences of bases in a gene that do not code for amino acids, and therefore they don't get coded for to create anything in the polypeptide chain. And in fact, they get removed out of the mRNA after it's been transcribed, and we call that splicing. The introns get spliced out of the mRNA. Exons are sequences of bases in a gene that do code for sequences of amino acids. So those are the coding sections on your mRNA. Then we have start and stop codons. And at the start of every gene, there is a start codon, meaning 3 bases. And those bases enable the ribosome to attach to the mRNA sequence, and that initiates translation. At the end of every gene, there are 3 bases that do not code for an amino acid, and that is the stop codon. And when the ribosome reaches those 3 bases, because it doesn't code for an amino acid, there's no corresponding tRNA molecule, and it causes the ribosome to detach from the mRNA, and therefore it ends translation.
Transcription is the first stage in protein synthesis, and this is the process in which a complementary mRNA copy of one gene on the DNA is created in the nucleus, and that can then leave the nucleus and be used in the next stage, translation. So let's go through what the key marking points would be for the process of transcription which happens in the nucleus. First of all, for our DNA molecule, hydrogen bonds are broken between the DNA bases, and that causes the double helix to unwind, like we can see here, and the 2 strands to separate. And one of those strands then acts as the template for the creation of the new mRNA strand. Then we have these 3 mRNA nucleotides within the nucleus, and they will align opposite the template strand according to their complementary base pairing. But bearing in mind that we have uracil instead of thymine in RNA. So if you have an adenine base, then the complementary base pair for that is uracil. The enzyme RNA polymerase is the enzyme responsible for catalyzing the joining of those mRNA nucleotides. The text is cut off a little bit here, but just a condensation reaction. So that joining together of those adjacent nucleotides is catalyzed by RNA polymerase. It's a condensation reaction, and it creates phosphodiester bonds. So that is how we create our mRNA molecule. And once it's copied, the mRNA is modified, and then it can leave the nucleus through the nuclear envelope pores.
Second stage of protein synthesis, which is translation. So that modified mRNA has left the nucleus through the nuclear pore, and then it attaches to the small subunit of the ribosome at the start codon. The tRNA molecule with the complementary anticodon to the start codon aligns opposite the mRNA, and that is held in place by the ribosome. And as we can see here, the ribosome can hold 2 tRNA molecules at a time, and once the peptide bond is formed, the previous tRNA molecule is detached, and the ribosome then moves along. So that's what we can see in that image. And forming that peptide bond requires an enzyme and also ATP. So the ribosome continues to move along the tRNA, enabling the next complementary tRNA anticodon to align to the next complementary codon on mRNA. And it continues until the ribosome reaches a stop codon, which causes the ribosome to detach, and it ends translation. That polypeptide chain that is now created then enters the Golgi body for folding and modifications. The next part of this topic is enzymes. Enzymes are biological catalysts made up of globular proteins. The active site, which we can see here, is a specific and unique shape due to the folding and bonding in the tertiary structure of a protein. Due to that specific shape active site, enzymes can only attach to substrates that are complementary in shape. So we describe them as being specific. Enzymes catalyze both intracellular and extracellular examples. And intracellular means inside of a cell, extracellular means outside of a cell. So for example, catalase is an intracellular enzyme inside liver cells that breaks down hydrogen peroxide into oxygen and water. Trypsin is an extracellular enzyme in the small intestines that hydrolyzes proteins. And the way that enzymes are catalyzing reactions is they are lowering the activation energy. So all reactions require a certain amount of energy before they occur. And that's what the activation energy is. Taking us into chemistry now. When the enzymes attach to the substrate, they lower that activation that is needed. And that is why the reaction speeds up. So what you need to be able to do is explain how enzymes lower the activation energy. And there's two different hypotheses that went about explaining this. The lock and key is the older model, and this model suggests that the enzyme is like a lock and that the substrate is like a key, and that key is perfectly complementary in shape, so it fits into the lock. So in this analogy, due to the enzyme's specific tertiary structure, it's completely complementary, so it can slot that substrate into the active site. And the theory here is stating that when we then get these enzyme-substrate complexes, the charged groups within the active site were thought to distort the substrate, and therefore it lowered the amount of energy required to break the bonds in the substrate. Now, since we've started to learn more about the molecular structure of proteins and understanding that they are actually slightly flexible and can move, that hypothesis was updated. So the induced fit hypothesis is the current accepted model. And this one suggests that an enzyme is like a glove and the substrate is like your hand. And by that we mean the glove and the hand are not perfectly complementary in shape until you put your hand inside the glove. So the induced fit model is when the enzyme active site is induced, meaning it's caused to change shape around the substrate. So initially, the substrate and the active site aren't perfectly complementary in shape. But when the substrate collides into it, it induces that active site to mold around the substrate, and then it does become perfectly complementary. Now, when that happens, that enzyme-substrate complex occurring puts strains on the bonds in the substrate, and therefore it lowers the activation energy required to catalyze that reaction. Now, because enzymes are globular proteins, they are very sensitive to certain conditions, and the following conditions can affect the rate of enzyme-controlled reactions: temperature, pH, enzyme concentration, and substrate concentration.
One really important edit just to be aware of, as I go through all of these variables now, such as temperature that you're about to see here, but also to do with substrate enzyme concentration, you need to say the frequency of successful collisions increases, and frequency is key for the mark scheme. It's not going to say that on all of the slides. I've edited this in right now for the 2025 video, but just be aware that you have to say frequency of successful collisions increases to get the marking points.
So if we start with temperature, we can see this particular shape curve. There is an increase in rate with temperature. We reach our optimum, and then there is a sudden decrease in rate at higher temperatures. And the explanation for this is at lower temperatures there is less kinetic energy, and therefore you are less likely to have a successful collision between the enzyme and the substrate. However, at higher temperatures there is now so much kinetic energy it can start to cause lots of additional movement, and those high temperatures will start to break the bonds in the tertiary structure. So for example, the hydrogen bonds are going to be breaking. That causes 3D shape to unfold, and you lose that unique shape active site. Now, the Q10 temperature coefficient is a measure of the rate of change of an enzyme-controlled reaction as a result of increasing the temperature by 10 degrees C. And the formula to work this out is R2 divided by R1. R1 is the rate of reaction at a temperature of and your X temperature is whatever your first temperature is. R2 is the rate of reaction at a temperature 10 degrees higher than the one you're comparing it to. Next then we have a look at the pH, and too high or too low a pH will interfere with the charges in the amino acids in the active site. That will cause the ionic and the hydrogen bonds to break, and therefore that unique 3D shape of the tertiary structure unfolds. That changes the shape of the active site and the enzyme denatures. Now, as indicated in this graph here, enzymes do have different optimal pHs though, depending on where they work. So, for example, any of the proteases digesting proteins that are found in the stomach, they actually have an optimum of around 1 or 2, so pH 1 or 2, because they are working in acidic conditions. Whereas some enzymes actually work better in slightly alkaline conditions, for example, trypsin and amylase, because in the small intestines it is slightly alkaline. We then move on to looking at the impact of enzyme and substrate concentration. Now, these do not cause the enzymes to denature or cause any change in shape to the enzyme. These have an effect based on the idea of saturation. So if there is a low concentration of substrate, the reaction will be lower as there will be fewer collisions possible between the enzyme and the substrate, because there are just fewer molecules there to potentially collide. If you were to increase the substrate concentration, because there are now more molecules present, you're more likely to have a collision, therefore more likely for enzyme-substrate complexes to occur, and the rate would increase. However, the rate would eventually plateau, so it would level off at a maximum rate of reaction, and that's because it would reach the point when all the enzyme active sites are in use, or in other words, the enzymes are saturated, and you would now have to add more enzymes as well as more substrate to increase the rate. Moving on to the enzyme concentration effect. At a low enzyme concentration, there will be a low rate of reaction. And that's because if there's fewer enzymes, there's fewer active sites for the substrate to bind to, and therefore there'd be fewer enzyme-substrate complexes. Increasing the enzyme concentration will increase the rate of reaction because there'll now be more enzyme-substrate complexes. But at high enzyme concentrations, unless unlimited substrate is added, the rate of reaction will plateau because there will be insufficient substrate to bind to the large number of enzymes, and you'd end up with lots of empty enzymes not being used. Competitive inhibitors also affect the rate of enzyme-controlled reactions, and there's two types of inhibitors we're going to look at. Competitive are the first type.
2025 edit that I'm adding in here to do with mark scheme specificity. For OCR, you do have to say that the competitive inhibitors are similar in shape, not the same. So they are similar in shape to the substrate. So make sure you are using the word similar in your answer.
A competitive inhibitor is the same shape or very similar in shape to the substrate. And that means it's complementary in shape to the active site, and it can actually bind to the active site. And that's what we're seeing here. This is the competitive inhibitor. It's binding to the active site. That forms an enzyme inhibitor complex instead of an enzyme substrate complex. It prevents the substrate from binding, and therefore it lowers the rate of reaction. Now, most competitive inhibitors are reversible, and reversible means that they can be removed from the enzyme. Whereas if it was non-reversible, that means it's permanently bound to that enzyme. Now, the importance or the relevance of that is, if you were to add in a much higher concentration of substrate, they would actually start colliding and banging into the inhibitor, knocking the inhibitor out, and therefore the substrate would then be able to bind. So with competitive inhibitors, at high concentration of substrates, they actually don't have any impact anymore. And that's what we can see here in this graph. At low substrate concentrations, the competitive inhibitor has lowered the rate of reaction compared to the enzyme reaction with no inhibitor, but at a high concentration, they both plateau at the same maximum rate. The non-competitive inhibitors though, these bind onto the enzyme at a position other than the active site, and we call that the allosteric site. Because the inhibitor is bound to the enzyme, it causes the protein to change shape, and therefore it changes the shape of the active site and the substrate can no longer bind, regardless of how much is added. And that's why we see there is a lower rate and even plateaus at a lower rate. And it doesn't matter if you continue to add more and more substrate.
Just interrupting here to add in another 2025 edit to make sure your answers are really mark scheme specific. For this bit here, you'd need to say that enzyme substrate complexes form less frequently. So that is the phrase that they'd be looking for. They're forming less frequently, and therefore the rate of reaction is much lower.
Because the active site is now a different shape, we get fewer enzyme substrate complexes. Now, some inhibitors are what we call end product inhibitors. And this is when the product of the reaction is a reversible inhibitor for the enzymes involved in controlling that reaction. And this is really useful because it enables reactions to be controlled. So essentially, it can turn reactions on and off. So if there is a lot of the product already present from this reaction, that product binds to the enzyme and inhibits it, and it prevents any more of that reaction happening. But when the product starts to run low, the inhibitor is no longer going to be able to inhibit the enzyme, and the reaction starts up again. So that prevents resources from being wasted. So that brings us on to the idea of coenzymes, cofactors, and prosthetic groups. Some enzyme-controlled reactions require an additional non-protein molecule, such as a coenzyme, cofactor, or a prosthetic group, to catalyse the reaction. So if we have a look at coenzymes and cofactors first, some reactions require atoms to be carried from one reaction to the next in a multi-step pathway of reactions, and that is actually the case in both respiration and photosynthesis. Some enzymes also require a non-protein molecule to bind to the active site to make it complementary to the substrate. And that is what a cofactor and a coenzyme is. The difference between the two is that coenzymes are organic molecules and cofactors are inorganic molecules, meaning they don't contain carbon. A prosthetic group on an enzyme is a type of cofactor, but they differ in that they are permanently attached to the enzyme by a covalent or a non-covalent force. Precursor activation is when enzymes often occur in an inactive form and they require to be activated by a cofactor so that they can actually work. And this prevents enzymes from causing damage within cells and it ensures they are only used when they are needed. An enzyme is activated by the binding of a cofactor, as this causes a change in the shape of the tertiary structure so that the active site now becomes complementary enough in shape to its substrate for it to bind. We then move on to biological membranes.
So biological membranes, all cells and all organelle membranes are composed of a phospholipid bilayer, and that's what we mean by a biological membrane. Plasma membranes provide this partially permeable membrane, and they're the site of chemical reactions, and they have a role in cell communication as well. And the plasma membrane has this model to represent the different components and properties, known as the fluid mosaic model. So the plasma membrane is described as a fluid mosaic model due to the movement of the phospholipids, proteins, glycoproteins and glycolipids, but also the arrangement or the pattern of the proteins within the phospholipids. The phospholipids align as a bilayer, which we can see here. We've got two layers of that phospholipid, and that's due to the hydrophilic heads being attracted to water, which are therefore on the outside of the membrane, and the hydrophobic tails being repelled by water, which is why they face inwards to each other. Proteins within the cell surface membrane can be extrinsic, which means on the outside, or intrinsic, which means going all the way through. The extrinsic or peripheral proteins provide mechanical support, or they can make glycoproteins and glycolipids, and the functions of those are in cell recognition as receptors. The intrinsic or integral proteins are the carriers or channel proteins, which are involved in the transport of molecules across the membrane. Protein channels, like we can see here, form tubes that fill with water to enable water-soluble ions to diffuse, whereas the carrier proteins will bind with ions and larger molecules such as glucose and amino acids. They then change shape to transport the molecule to the other side of the membrane. Finally, there's cholesterol, which we can see just here. That's present in some membranes, and this restricts the lateral movement of other molecules in the membrane. And this is useful as it makes the membranes less fluid at high temperatures, and this will prevent water and dissolved ions from leaking out of the cell or out of an organelle.
So these membranes are affected by certain factors. Temperature does have an effect on the structure and the permeability of that phospholipid bilayer. And there's two reasons why. Firstly, at high temperatures, it increases the kinetic energy of those phospholipids, so they're going to have even more movement. And that increase in the fluidity of the membrane means that there's going to be larger gaps periodically between the phospholipids, and therefore the permeability increases, and it makes it easier for particles to cross the membrane. The second reason is high temperatures could denature the carrier and the channel proteins in the membrane. And if that happens and the protein channels and carriers become wider, then even more molecules are going to be able to move across that cell membrane that weren't initially able to. So therefore, it becomes more permeable. Solvents can also damage the cell membrane structure. Organic solvents like alcohol, for example ethanol, dissolve lipids, so they will dissolve the phospholipid bilayer in the membranes. And that damage causes the fluidity of the membrane to increase and become more permeable. Next, we move on to movement across the membranes. And there are 6 key modes of transport in and out of cells that we're going to have a look at. And it's this 6 just here. So starting with simple diffusion. This is the net movement of molecules from an area of higher concentration to an area of lower concentration until equilibrium is reached. This process does not require ATP. For molecules to diffuse across the membrane, they must be lipid soluble and small. And that's what we can see here. We've got a high concentration on one side of the membrane compared to the other, and these lipid soluble molecules then dissolve in that phospholipid bilayer and move down their concentration gradient. And this will continue until there is no longer a concentration gradient present. Facilitated diffusion is still a passive process with the movement going down the concentration gradients. However, it's for molecules that are either too large to diffuse through the phospholipid bilayer or for molecules that are not soluble in lipids. And so instead they have to move through the proteins that are embedded within the phospholipid bilayer. So that movement of ions and those polar molecules would be through either protein channels or the protein carriers. Osmosis is the movement of water from an area of a higher water potential to an area of lower water potential, or more negative. And it happens across a partially permeable membrane. And the more negative a water potential, that means the more concentrated the solution. You have less water but more solutes dissolved in it. And there's different types of solutions that occur that have an impact on osmosis. An isotonic solution is when the water potential of the solution is the same in the solution and the cell. And because there is no water potential gradient, there'd be no net movement of water in or out of a cell. And in an animal cell, we can see here is a red blood cell as an example, that has no impact on the overall size or shape of the cell. A hypotonic solution is when the water potential of the solution is more positive, so it's closer to 0, which basically means there is more water compared to the solute. So as a result, water will move from the solution into the cell, causing it to swell, and it could even eventually cause it to burst if enough water is moved in. In a hypertonic solution, that means the solution is more negative than the water potential in the cell. So as a result, water within the cell will move out by osmosis into the solution, and that causes the cell to shrivel up. But the term we use for that in animal cells is crenation, and we say in plant cells they are plasmolysed. In plant cells as well, they will not burst as readily because they have that cellulose cell wall. So instead they become really swollen and we describe it as turgid. The next type of transport is active transport. And this is the movement of molecules and ions from an area of lower concentration to an area of higher concentration, which means it's going against the concentration gradient. And for this reason, it requires energy in the form of ATP from respiration. And it involves carrier proteins. It is a selective process as only certain molecules are able to bind to a receptor site on the carrier proteins. When they do that, ATP will bind to the protein on the inside of the membrane. It then gets hydrolyzed into ADP and PI. That causes the carrier protein to change shape and open towards the inside of the membrane. And as a result, the molecule that was attached is released to the other side of the membrane. The phosphate group, or that PI molecule, is then released from the protein, and the protein reverts to its original shape, and the process can continue to happen as long as ATP is present. The next one is endocytosis, and this is a type of active transport, but it is the bulk transport of molecules into a cell. The cell surface membrane bends inwards around the molecules surrounding it to form a vesicle. And that's what we can start to see happening here. It's bending inwards and eventually it folds all the way round to form a vesicle. The vesicle pinches off and moves within that cytoplasm. And endocytosis can be classed as either phagocytosis, as we've got on the left, or pinocytosis that we have on the right. And when it's a solid particle being taken in, it's called phagocytosis. When it's a liquid being taken in, that is when it's pinocytosis. This requires energy from ATP because to change the shape of the membrane around that material does require energy. Exocytosis is another type of bulk transport, but this time it's the movement of molecules out of a cell. So the molecules are contained with a vesicle. Vesicles move towards the cell surface membrane, fuse with the membrane, and the contents of that vesicle is then released to the outside of the cell. This process requires energy because ATP is needed to move the vesicle along the cytoskeleton towards the cell surface membrane. Cell division, cell diversity, and cellular organization. So cell division, first of all, in eukaryotic cells, they enter the cell cycle and then divide by mitosis or meiosis. Prokaryotic cells replicate by binary fission, and then viruses do not undergo cell division as they are non-living. The cell cycle comprises 3 key stages. We have interphase, split into G1, S phase, and G2. Then there's the nuclear division, which is either mitosis or meiosis, and then finally cytokinesis. So let's begin with interphase, and this is the longest stage of the cell cycle. G1, which is the first part of interphase, is when protein synthesis occurs to make proteins involved in synthesizing organelles. The organelles then replicate. The cell is checked that it is the correct size, has the correct nutrients, growth factors, and that there is no damaged DNA. If a cell doesn't pass these checks, then replication does not continue. Then we have S phase, and this is when DNA is replicated. G2 is when the cell continues to grow, energy stores increase, and the newly replicated DNA is checked for any potential copying errors.
I've edited this slide for 2025 to make sure that the descriptions of what's happening in G1 and G2 are really mark scheme specific. So make sure you have a look at the level of detail on this slide when you're making your notes or flashcards, because this is the level of detail required for the mark scheme.
We'll then move on to the first option for nuclear division, which is mitosis.
One quick edit I've made here is to make sure it's really mark scheme specific. For mitosis, you have to say it creates 2 genetically identical diploid cells. So you would have to say genetically identical to get the mark.
And it's used for growth, tissue repair, and asexual reproduction in plants, animals, and fungi. There are 4 key stages: prophase, metaphase, anaphase, and telophase, or PMAT, as a way to try and remember it. Prophase is when the chromosomes condense and become visible. And in animal cells, The centrioles, which are shown here in yellow, separate and move towards the opposite poles of the cell. The centrioles create spindle fibres, which are released from both poles to create a spindle apparatus. And these will attach to the centromere and the chromatids on the chromosomes in late prophase, early metaphase. Plants have spindle apparatus, but they don't actually have the centrioles. Metaphase is the next stage, and this is when those spindle fibres have attached to the centromere and it causes the chromosomes to align along the equator of the cell. The spindle assembly checkpoint also occurs in this stage, and this is where there is a check to make sure that every chromosome has a spindle fibre attached to its centromere before mitosis carries on to the next stage, which is anaphase.
So in anaphase, this is when the spindle fibres start to shorten and move towards the centrioles and pull the centromere and chromatids they are bound to towards those opposite poles as well. This causes the centromere to divide into two, and the individual chromatids are then pulled to the opposite pole. This stage requires energy in the form of ATP, which is provided by respiration in the mitochondria.
Next we have telophase or telophase, and the chromosomes are now at each pole of the cell and become longer and thinner again, so therefore they start to no longer be visible. The spindle fibres will disintegrate and the nuclear membrane starts to reform around those chromosomes. Cytokinesis is then the final stage of the cell cycle, and this is when the cytoplasm splits so we get two genetically identical cells. And in animals, the way this happens is a cleavage furrow forms in the middle of the cell and the cytoskeleton causes the cell membrane to draw inwards until the cell eventually splits in two. In plant cells, the cell membrane splits into two new cells due to the fusing of vesicles from the Golgi apparatus. The cell will form new sections around the membrane to complete the division into two cells.
Just interrupting here to point out a change to this slide to make sure your answer is really mark scheme specific. You have to say that it causes the cell surface membrane to draw inwards and the cell surface membrane splits, not cell membrane, it's specifically the cell surface membrane. So you have to say that word surface.
Now you can actually observe mitosis, and this is one of the required practicals. And the stages of mitosis can be viewed using a light microscope in onion or garlic root tips. So you would need to take a thin slice of the root tip from either an onion or a garlic and place it on top of a microscope slide. You then break it down a little bit with a mounted needle. A stain is then added, and the purpose of this is to make the chromosomes visible when you put your slide under the microscope. You would then sometimes add acid to help to break down the cellulose connections between the cell walls. Then you would place a cover slip on top and push down. The reason we push down is to squash the tip to achieve a single layer of cells to ensure light can pass through, and therefore you can see the individual cells and the chromosomes inside of them. And that's what we can see here, that single layer of cells, light's passing through them. Most of the cells here are in interphase, and we can tell that because chromosomes are not visible. But we do have two where the chromosomes are visible. So we could use this to calculate the mitotic index. So this is calculated by counting how many cells are visible in the field of view, meaning the section that you're looking at, and by counting the number of cells that are currently in mitosis. And it's essentially a percentage, this. So the number of cells in mitosis divided by the total number of cells, and then you'd multiply it by 100 to give that answer as a percentage. Now, when they have questions like this, they usually tell you the total number of cells so that you don't end up spending lots of time in an exam just counting lots of cells, and instead you just have to work out the number of cells in mitosis. And like I said, in this example, we've got two that are in metaphase. So the other type of nuclear division is meiosis. And this is when we have two nuclear divisions, and that results in four genetically different haploid daughter cells. The two rounds of division are referred to meiosis 1 for the first round and meiosis 2 for the second. Both stages include prophase, metaphase, anaphase and telophase, and cytokinesis at the end. but interphase only happens at the very beginning before meiosis 1. Now we said it makes a haploid cell, and what we mean by haploid and diploid is a haploid cell, represented by a single letter N, is when you have one copy of each chromosome. A diploid cell, or 2N, is when you have two copies of each chromosome. The genetic differences are introduced in meiosis by two key processes: independent assortment of homologous chromosomes and crossing over. So let's have a look at crossing over first. During prophase 1, the homologous chromosomes pair to form bivalents. And by bivalent, we mean two homologous chromosomes next to each other. And that's what we can see here. We've got our homologous chromosomes represented as one in red, one in green. And they look like this X structure because the DNA is replicated. So we have two sister chromatids attached by a centromere to make a chromosome. And here is the homologous chromosome. Crossing over genetic material can occur. So these non-sister chromatids, meaning a chromatid from the different chromosomes, crossing over and form what we call a chiasma, and that is where the crossing over happens. Now the tension that creates can result in breaks occurring, and we then get this exchange in material of those chromatids. And as a result, the alleles that were on this chromatid are now going to be part of this chromosome. So we have this new combination of alleles in the resulting gametes. Independent assortment also increases the genetic diversity, and this happens during metaphase 1, where the homologous pairs of chromosomes line up opposite each other on either side of the equator. But it is random for each homologous pair which side the maternal and the paternal chromosomes align. And that's what we can see here. We've only been shown an example with 3 homologous pairs, but it shows you all the possible combinations of how they could align at the equator. And for humans, we have 23 homologous pairs. So that means there would be 2 to the power of 23 possible combinations of how those homologous pairs could align. And that works out to over 8 million different combinations. And that's what we can see here. And you can use that formula, 2 to the power of n, where n is the number of homologous pairs, to work out the number of possible combinations for any different organism.
Now in metaphase 2, the sister chromatids within one chromosome also are lining up at the equator, and their orientation on each side of the equator introduces another chance for increased genetic variation.
And as a result, each gamete receives different combinations of the maternal and paternal chromosomes. We then move on to this idea of organisation, looking at specialised cells as well. Now, multicellular organisms are organised in the following way. Cells are the smallest structure, then we get tissues, organ systems, and those all work together to create the entire organism. Now, you do need to be aware of a selection of specialised cells. And we've got here the summary of all of the ones that you need to know about. So I recommend that you take a screenshot of this, make your own table, make your own notes, or even turn this into a flashcard. Because you've got the 5 specialised cells, and for each one, you've got your description here of the structure of the cell and how that links to the function.
Now I've added this into the 2025 video because you do actually also need to know about root hair cells. So these are added in here. So these are cells on the surface of the roots, and they have long projections to increase the surface area for osmosis of water and also the active transport of mineral ions from the soil. They have a thin cell wall reduced to diffusion distance.
We then move on to the different tissues. Same thing again, screenshot it in print, have it in your notes or copy it out, or even better, turn it into flashcards.
Just to point out a key edit here, these are tissues, so I've edited the slides that you're going to see coming up, which said epithelial cells and ciliated epithelial cells. However, these are the tissues, so at this point you would just say a squamous epithelia tissue and ciliated epithelia tissue. But those are made up of ciliated epithelial cells. But it's really important that when you're describing the tissue, you don't put cell at the end.
These are the different tissues that you need to be aware of, the structure of each, and the function of them. We then move on to stem cells, and these are undifferentiated cells that can self-renew, meaning continually divide and become specialized. Different types of stem cells have different differentiation abilities. And the types that you need to know about are totipotent, pluripotent, multipotent, and unipotent. Totipotent cells can divide and produce any type of body cell. During development, totipotent cells translate only part of their DNA, resulting in cell specialization. Totipotent cells occur only for a limited time in early mammalian embryos. They then develop into pluripotent stem cells, and these are found in embryos and can become almost any type of cell. The only cell that they can't form is the placenta. So for this reason, they are used in research with the prospect of using them to treat human disorders. There are issues with this though, as sometimes this treatment doesn't work, or the stem cells can continually divide and that results in tumours. On top of those practical issues, there are ethical issues as well, because there's a debate as to whether it's right to make a therapeutic clone of a patient in order to create an embryo genetically identical to them, to get the stem cells to cure a disease, and then after that, destroying the embryo as well. So the other two types of stem cells are multipotent and unipotent, and these occur in mature mammals, and they can only divide into a limited number of types of cells. Multipotent cells, such as the ones found in bone marrow, can differentiate into a limited number of cells, and in bone marrow that is into the different types of blood cell. Unipotent cells can only differentiate into one other type of cell. So the potential uses of stem cells are in both research and medicine, and these include potentially being used to repair damaged tissues, or in the treatment of neurological conditions such as Alzheimer's and Parkinson's, or it could be research into developmental biology. So here are the three key sections that this topic is split into, and we're going to begin with the exchange surfaces. Now this starts with knowing about the importance of surface area to volume ratio. And exchange surfaces in organisms have many similar adaptations, and that is to make sure that substances such as oxygen and carbon dioxide can exchange across the surfaces as efficiently as possible. Smaller organisms like amoeba, which we have in this diagram here, have a very large surface area compared to their volume. And that means that, first of all, they have a big surface area for the transport of substances, but also it means there is a short distance between the outside and the very middle of the organism. And for that reason, they don't require any special adaptation organs or systems for the transport, and instead simple diffusion is sufficient to meet their metabolic needs. Now in contrast, larger organisms have a smaller surface area compared to their volume, and that means there is a larger distance from the very outside of the organism to the very centre. Now on top of that, larger organisms also have higher metabolic rates, meaning that they will require more oxygen for respiration to be able to create ATP. Also, as we said, they've got a smaller surface area to volume ratio, and they have got a longer distance from the outside to the middle of the organism. And therefore, they require adaptations to increase the efficiency of exchange across their surface. And this then takes us into this topic where we start off by looking at the gills in fish, the alveoli in humans, and the tracheal system in insects as adaptations. So the running theme throughout this topic is looking at what provides the large surface area, what helps to maintain a concentration gradient, and what reduces the diffusion pathway. So those will be the adaptations that we'll focus on in the fish, the insects, and in the humans. Now in general, to increase the surface area, you're looking for structures where you might have projections, so that's what you see in root hair cells, the long protruding part of the cell, or it could be folded membranes that also increases the surface area. Concentration gradients, that can be maintained through ventilation or a good blood supply, removing the blood which contains high concentrations of substances, And in fish, we're going to have a look at the countercurrent flow mechanism. And then the length of the diffusion pathway can be reduced, mainly by having only a single layer of cells, and that layer is normally squamous epithelial cells. So those concepts we're going to have a look at in a bit more detail, starting with the mammalian gas exchange system. And the structures that you need to be familiar with are the trachea, bronchi and bronchioles, and the alveoli. So let's start with the trachea, also known as the windpipe. And the trachea has these C-shaped rings of cartilage that run all the way down. And that is to support the trachea to make sure that it doesn't stick together and close. So it stays permanently open so air can flow through. It's also lined by epithelial cells which are ciliated and contain goblet cells. Ciliated cells are those hair-like structures that help to sweep away any mucus in the trachea. And goblet cells are the cells that make that mucus. So the mucus is really thick and sticky, and therefore any pathogens, dust particles will stick to the mucus, and then the cilia will sweep that mucus up the trachea so it can be coughed out, and therefore it doesn't reach the lungs and cause any potential infections. There's also smooth muscle within the walls of the trachea, and that muscle can contract if there are any harmful substances within the air. And that results in the lumen, so this space in the middle here, of the trachea constricting. That reduces the airflow to the lungs. Now, when the smooth muscle relaxes, the lumen dilates, and that stretch and recoil of the lumen is possible because there is also elastic fibres within the tracheal walls. The bronchi and the bronchioles are the next structures that you need to be familiar with. The trachea, which we can see just finishing here, in humans, it then splits into two tubes, which are the bronchi. Singular would be bronchus. And that is to connect to both the right and the left lung. Now, those split even further into these smaller tubes, which are the bronchioles. And at the end of the bronchioles, we then have the alveoli. Both the bronchi and bronchioles also have cartilage within their walls, and that is to provide structural support to keep those tubes open. Lastly, then we get to the alveoli. And as I said, these are located at the end of the bronchioles, and this is the site of gas exchange. Oxygen from the alveoli will diffuse into the blood and will be picked up by the red blood cells. And carbon dioxide within the blood in the capillary is going to diffuse into the alveoli and then be exhaled. So this then brings us to those three features that you always need to comment on in this topic. What provides the large surface area, the short diffusion distance, and what maintains a concentration gradient. The large surface area is provided by the fact there are a very, very large number of alveoli in both sets of lungs. So one alveoli doesn't have a large surface area. It's the fact that there are millions of those alveoli. The short diffusion distance is because the alveoli walls, and actually the capillary walls as well, are both only made up of a single layer of cells. And those are squamous epithelial cells, which means they're long and flat.
Lastly, to maintain the steep concentration gradient, each alveolus is surrounded by a network of capillaries. This means that once oxygen has diffused from the alveoli into the blood, it can be quickly transported away. And also the fact that there is ventilation occurring in the lungs means that carbon dioxide that is diffused from the blood into the alveoli is also quickly taken away. And ventilation is the mechanism of breathing which involves the diaphragm and antagonistic interactions between the external and internal intercostal muscles, bringing about pressure changes in that thoracic cavity. Now, the importance of this ventilation is it maintains a steep concentration gradient for gas exchange at the alveoli.
So when you inhale or inspiration, that is because there is an increase in the volume of the thorax, and that means this space here. So you need to know what causes that increase, and we'll come to that shortly. But the fact that there's an increase in volume means that the pressure will decrease, and because inside of the thorax there is now a comparatively lower pressure compared to the atmosphere, air flows into the lungs. When you exhale or expiration, there's a decrease in volume of the thorax. That increases the air pressure and it forces the air out of the lungs.
During forced expiration, the internal intercostal muscles contract, but when it is relaxed expiration, the internal intercostal muscles remain relaxed.
So the cause of those changes is the different muscles contracting and relaxing. So when you inhale, the diaphragm contracts. That causes it to move down and become flatter. The external intercostal muscles will contract and the internal ones relaxed, and that will pull the rib cage up and out. And that is what provides a larger volume. When you exhale, it's the exact opposite. The diaphragm relaxes and that causes it to dome upwards. And the external intercostal muscles will now relax and the internal intercostal muscles contract. And that pulls the rib cage inwards and down, reducing the volume of the thorax. So you can actually measure the volume of air inhaled and exhaled using a spirometer. And when you do that, you can get a graph that looks something like this. And this is labelled to show you what all of those peaks and troughs are representing. And we're shown here at the beginning, this is just normal respiration. So we're going to have some normal breathing. Then we have forced respiration, which is referring to a really deep inhale and exhale, and then it's going back to normal. And this bit here on this side is telling you what each peak and trough is showing. So your vital capacity, which we can see is going from this point to this point, that is the maximum volume of air an individual can inhale and exhale during a deep breath. Your tidal volume is the air inhaled and exhaled when you are at rest. The residual volume, which is down here at the bottom, that is the volume of air that always remains in the lungs so that the lungs don't ever fully empty and collapse inwards. And then to work out your breathing rates, you could use this graph to look at the number of breaths per minute. And we could do that by counting repeating patterns. So how many times we've got peaks over a particular period of time. You can also then work out ventilation rate, and that would be your tidal volume times your breathing rate. Oxygen uptake will increase when the ventilation rate increases, and that would happen during exercise, for example. Next, we move on to ventilation and gas exchange in fish, which also count as a large organism. Now, the challenge that fish face in particular is that there is less oxygen dissolved in water than you have oxygen in the atmosphere. So that's going to lead to issues with maintaining the concentration gradient. But first, we're going to have a look at the ventilation in fish. Now, fish swim with their mouth open so that water can flow in and over the gills, and the gills are the site of gas exchange. The fish will lower their buccal cavity, which is within their mouth, open their mouth, and that will increase the entire volume of the buccal cavity. And because there is now a larger volume, the pressure decreases, and that's what causes water to flow in. Simultaneously, the operculum valve, which we can see here as closed, is then going to shut, and the operculum cavity will therefore expand, which we can see in this section. That will cause an increase in the volume of the operculum cavity and therefore a decrease in pressure. The fish will then raise the floor of their buccal cavity, and that then forces the water from the buccal cavity over the gills and then out of the operculum. And at that point, when they've raised the buccal cavity and closed their mouth, the pressure is high and it opens the valve, and that is when the water can then flow over. So this ventilation ensures there is a constant flow of water over the gills for gas exchange. So the gas exchange in the fish happens over the gills, and there are 4 layers of gills on both sides of their heads. The gills are made up of gill filaments, which are these longer parts sticking out. And then every gill filament is covered in gill lamellae. And if we zoom in here, we can see the gill lamellae are these semicircle structures on top of the gill filaments. So if we think about those 3 features that we always have to look for, there is a large surface area because there are so many gill filaments which are all covered in gill lamellae. And the gill lamellae is the exact location of gas exchange. The short diffusion distance is because the gill lamellae and filaments are both very, very thin, and the gill lamellae contains this network of capillaries. So it's a short diffusion distance.
2 things I just want to note about this slide, which I've updated for the 2025 video, which you're now watching, is you don't actually need to know gill plates, so that has now been removed. And the mark scheme is really specific that you have to say maintains a steep concentration gradient, not just maintain a concentration gradient.
Maintaining the concentration gradient links to the final and the biggest focus, which is the countercurrent flow mechanism. The countercurrent flow mechanism is to compensate for the fact that water has a lower dissolved oxygen concentration compared to the oxygen concentration in the atmosphere. For fish to be able to maintain the concentration gradient for diffusion across the entire lamellae and therefore maximize diffusion, the countercurrent flow mechanism has to be used. And this is when water flows over the gill lamellae in the opposite direction to the flow of the blood in the capillaries. And that's what we can see here in the diagram. We've got in one direction, we've got the flow, and then the opposite direction, the other flow. So we would have here, the top one would be representing the water, and the bottom one would be representing the blood. And because they're flowing in opposite directions, an equilibrium in the concentration of oxygen is never reached, and that is why diffusion can be maintained along the entire gill lamella.
You may have noticed as well, this is another 2025 update I'm just slotting in at this point, and that is steep concentration gradient for diffusion is essential not just to maintain the concentration gradient.
We then move on to gas exchange in insects, and terrestrial insects have a tracheal system, which is made up of spiracles and trachea and tracheoles. The spiracles are valve-like structures that run along the side of the abdomen, and they're very much like the stomata in plants. So they can open and close to allow gases to move in and out, but it's also to help prevent water loss. Now those spiracles are attached to trachea, and those trachea branch into many branching tracheoles, and the tracheoles are the site of gas exchange in insects. So how then we have the large surface area, the short diffusion distance, and the maintained concentration gradient are due to these facts we've got here. So first of all, the insects can contract and relax the muscles they have in their abdomen, and this creates this pumping mechanism. So gas gets pumped in and pumped out, a bit like the idea of ventilation. On top of that, the large surface area is maintained by many branching tracheoles, and the fact that there's so many is providing the large surface area. There's a short diffusion distance, again because there are so many branching tracheoles, they will reach a really wide distance across the abdomen of the insect, and the wall of the tracheoles are really thin as well. The concentration gradient is maintained by the cells respiring, so they're going to be using up oxygen and producing carbon dioxide, and that is going to create the gradient. But also when the abdominal muscles contract, it pumps new air in and old air with lots of carbon dioxide in, out. And when insects are in flight, we get an added effect because the muscles are going to be contracting and relaxing more rapidly, and that will result in them starting to respire anaerobically and producing lactate. And that lactate will dissolve to form lactic acid, and that will lower the water potential of the cell. Now this causes water, and there is always some residual water within the tracheoles, which we call tracheal fluid, to move into the abdominal cells by osmosis. This decreases the volume of liquid inside of the tracheoles, and because now there's less liquid in that same space, it causes a decrease in the pressure, and therefore air from the outside will move in through the spiracles.
At this point, there's another addition to the slide for 2025, and that is maintaining a steep concentration gradient. So as you can see, the mark schemes are now really specific. You have to emphasize steep concentration gradient.
3.12 Transport in animals. So we begin this by looking at circulatory systems, and each animal has a circulatory system adapted to meet its needs. All circulatory systems will transport gases, nutrients around an organism in a transport liquid, for example the blood, and the liquid is transported around in vessels, and there's usually a pump as well to help move that liquid, for example the heart. So we're going to take a look at these 4 types of circulatory systems, starting with the open compared to the closed. So an open circulatory system is what you'd see in invertebrates like insects, and the transport medium, the hemolymph, is usually pumped directly to the body cavity called the hemocoel. and there are very, very few transport vessels. The transport medium is pumped at low pressure, and it will transport food and nitrogenous waste, but not gases, which are instead transported via the tracheal system, which we just saw. Once exchange has taken place at the cells and tissues, the transport medium returns to the heart through an open-ended vessel. Now, in contrast, the closed circulatory system, this is what all vertebrates would have, and some invertebrates, such as annelid worms. And in this instance, the transport medium is blood, and it always remains inside of blood vessels. Gas and small molecules can leave the blood by diffusion or due to high hydrostatic pressure, and that's what we'll look at in tissue fluid formation. The closed circulatory system transports oxygen and carbon dioxide, and the oxygen is usually transported by pigmented protein, for example, hemoglobin. So next then, looking at the single closed versus double closed circulatory systems. In a single circulatory system, this means the blood passes through the heart once per cycle, and there is only one circuit that the blood takes. For example, fish have single closed circulatory systems. The blood passes through two sets of capillaries. Immediately after being pumped out of the heart, the blood then flows through capillaries in the gills to become oxygenated. The blood will then flow through capillaries, delivering the blood to the body before returning it back to the heart. This system would not enable efficient gas exchange for mammals, but it does work for fish because they have that countercurrent flow mechanism. So the double closed circulatory system, the blood passes through the heart twice per cycle, and there are two separate circuits the blood would take. Now we see this in birds and most mammals. And one of the circuits is blood vessels carrying blood from the heart to the lungs for gas exchange. And the second circuit is blood vessels carrying the blood from the heart to the rest of the body to deliver oxygen, nutrients, and collect the waste.
The last thing to note here on the circulatory systems is you do actually need to know the names of these two circuits. So the one circuit that carries the blood from the heart to the lungs is the pulmonary circuit, and the other circuit where it carries the blood from the heart to the rest of the body is known as the systemic circuit.
So the blood vessels to be aware of then we have in this table: the arteries, arterioles, capillaries, venules and veins. There's a lot of information on this slide, and what I'd recommend is to pause it at this point, either take a screenshot or copy down this table, read through it in your own time so we can see the different structures that are present, and you've got some explanation as to why it's important. So this would also be really good to turn into a selection of flashcards. So if we have a look at the capillaries first, the capillaries form capillary beds, which is when you have many branched capillaries all connected. And this is typically at exchange surfaces, such as the outside of the alveoli. Capillaries have a really narrow diameter, and that is to slow down the blood flow. And the red blood cells can only just fit through that, and that is so that they end up being squashed against the walls, and this maximizes diffusion. Capillaries are also only made up of one single layer in their endothelium, and those are squamous epithelial cells. They're really squashed and flattened. That property of having only a single layer of cells and the fact that there are small gaps between those cells is what enables tissue fluid to form. Because of those small gaps, liquid and small molecules can be forced out of the capillaries due to high pressure, and this then forms tissue fluid. And it's called tissue fluid because it's a fluid that ends up surrounding tissues, the cells. So hydrostatic pressure, first of all, this is the pressure exerted by a liquid. Oncotic pressure is the tendency of water to move into the blood by osmosis. So tissue fluid formation and reabsorption is all centered around the interaction of hydrostatic and oncotic pressure. So let's look at the formation first of all. So as blood enters the capillaries from the arterioles, which is this end here on the diagram, this is a capillary, and there would be an arteriole here where the blood is leading into. The arteriole has a wider diameter compared to the diameter of the capillary, and the same volume of blood is therefore being forced into a smaller space. And this results in a high hydrostatic pressure. Now, because the hydrostatic pressure is high at this arterial end of the capillary, it forces out water and small molecules, such as glucose, amino acids, fatty acids, ions, and oxygen. Now, that liquid that is forced out, all of these molecules here, is then called tissue fluid because it is a liquid that is bathing the tissues. And this is actually an advantage because all of those substances that were forced out can then move into the cells if they need it. And any waste products that are made by these cells will move out and can then be picked up and transported away. So the hydrostatic pressure is higher than the oncotic pressure at this arterial end of the capillaries. And that's why the net movement is out of the capillaries and to form the tissue fluid. If we then have a look at the reabsorption of that water, large molecules aren't forced out of the capillaries because they're too big. So things like soluble plasma proteins will remain in the capillaries in the blood, and this will lower the water potential of the blood. So we therefore have a lowered water potential, which means a higher oncotic pressure. And this is moving towards the venial end of the capillaries. And at this point, the hydrostatic pressure has decreased because so much liquid has been forced out. So as a result, there is a net movement of liquid back into the capillaries by osmosis at the venial end. But once equilibrium has been reached in that water potential, because water is moving back in by osmosis, there can be no more water from the tissue fluid being reabsorbed into the blood. And this is why the final parts of the tissue fluid get absorbed into the lymphatic system. And as it moves into the lymphatic system, we then call it lymph. And lymph is very similar to plasma, except it doesn't contain those large plasma proteins and some of the blood cells. Next, we have a look at the mammalian heart. And this is an organ made up of cardiac muscle, and it's responsible for pumping the blood around the body inside of different vessels. The cardiac muscle is myogenic, which means it automatically contracts and relaxes, and it will never fatigue, which is a key difference to skeletal muscles. It has coronary arteries that are run on the outside, and these supply the heart muscle, or the cardiac muscle, with oxygenated blood for aerobic respiration. And that then means ATP can be created so that the cardiac muscle can continually contract and relax. Finally, the heart is surrounded by pericardial membranes, and these are inelastic membranes that prevent the heart from filling and swelling with blood. If we go into a little bit more detail then about the internal structures of the heart, the left ventricle has a thicker muscular wall, so it can contract with more force and pump blood at higher pressure. And that is needed so that the blood can flow out of the left ventricle, through the aorta and to the rest of the body, and it ensures that the blood will reach all of the different blood vessels so that all of the body can receive oxygenated blood. The right ventricle has thinner cardiac muscle in its wall, so it has less cardiac muscle, and that's because it doesn't need to contract with as much force because the blood is being pumped up and out of the pulmonary arteries to the lungs, and the lungs are much closer, so it doesn't need that high force. But the other reason is the blood needs to flow through the lungs at lower pressure so that it doesn't cause damage to the capillaries in the lungs by the alveoli, and so the blood flows slowly so there's more time for gas exchange. The atria are the two chambers at the top, and we can see here that the cardiac muscle is much thinner, and that's because they don't need to contract with much force at all because they are pushing the blood from the atria to the ventricles, which is very, very close. And also it's moving down with gravity. So then if we have a look at the cardiac cycle, it can be split into three key stages: diastole or diastole, depending how you pronounce it, atrial systole or systole, and ventricular systole or systole. Different people pronounce it different ways, but it means the same thing. Atrial systole is when the atria are contracting. Atrial diastole is when the atria are relaxing. Ventricular systole is when the ventricles contract, and ventricular diastole is when the ventricles contract. So if we start here on the diagram, at this point we have both the atria and the ventricles relaxing. They're both in diastole. And because those muscles are relaxing, we've got a larger space or larger volume in the chambers. Therefore, the pressure drops and the blood is going to flow into the two atria. Now, as the blood flows in, that starts to increase the pressure, and it increases the pressure high enough that it will force open the atrioventricular valves. We then get to this stage where the atria contract in atrial systole, and that will then cause that contraction and force the blood from the atria into the ventricles. After that point, the atria will then relax, and now the ventricles contract. And because the ventricles are now contracting, there is a higher pressure in the ventricles compared to the atria, and that causes the atrioventricular valves to shut. But when the pressure is high enough from that contraction, the semilunar valves will open, and this is what causes the blood to be pushed from the ventricles out of the pulmonary artery and the aorta. So the summary of that key information then we can see on the next few slides.
Now, unlike on this diagram, you don't actually have to know it's split into atrial diastole and ventricular diastole. You just need to know it as diastole as a whole for OCR. And this is the stage when the atria and ventricular muscles are relaxed. This will then result in the blood entering the atria via the vena cava and pulmonary vein. And the blood flowing into the atria increases the pressure within the atria, and that causes the atrioventricular valves to open so blood can begin to flow into the ventricles. The key points for atrial systole: this is when the atria muscular walls contract, increasing the pressure further. This causes the blood to flow into the ventricles through those open atrioventricular valves. And at this point, the ventricular muscle walls are relaxed.
After a short delay, the ventricles then contract, and that increases the pressure beyond that of the atria, so the atrioventricular valves close and the semilunar valves open. Now, this is a key math skill that comes up linked to this topic: cardiac output. And that is the volume of blood which leaves one ventricle in 1 minute. It can be calculated using the formula heart rate times stroke volume. And the heart rate is how many beats there are per minute, and the stroke volume is the volume of blood that leaves the heart each beat, typically in decimeter cubed. Next, we need to have a look at what is controlling the cardiac cycle, and by that we mean what is controlling when the atria and the ventricle contract and relax. So first of all, cardiac muscle is myogenic. It contracts on its own accord, but the rate of contraction is controlled by a wave of electrical activity. So the sinoatrial node, or the SAN, this is located in the right atrium and it's known as the pacemaker. And I've shown this here in bright yellow. The atrioventricular node, the AVN, is located near the border of the right and the left ventricle, so just here. The bundle of His runs through the septum, which is this section. And then we have the Purkinje fibres, and those branch into the walls of the ventricles. So these structures are all key in the control of the cardiac cycle. So the first thing that happens is that SAN releases a wave of depolarization across the atria. So that is going to spread from the SAN across the two atria. The AVN releases another wave of depolarization once that first wave reaches it. But there is a non-conductive layer between the atria and the ventricles, and that prevents that wave of depolarization spreading directly down the ventricles, and instead it forces it to move down the bundle of His, because that is the part that is conductive. And once it moves down the bundle of His, it then moves up through the Purkinje fibres. Now this is an advantage because as a result, it causes the apex, so the very bottom here, of the heart and the walls of the ventricle to contract, and there is a short delay before this happens. And that leaves enough time for the atria to have fully contracted and pumped the blood into the ventricles before the ventricles contract. So it's going to be a more efficient way to move the blood out of the heart. The final step we have is the diastole, and this is when repolarization occurs and the cardiac muscle relaxes. The electrocardiogram, or ECG, is a machine that's able to measure these waves of depolarization. So we can measure it using that ECG, and we can interpret it to diagnose any irregularities in heart rhythms. The ECG doesn't directly measure the electrical activity of the heart, but instead the differences in electrical activity in your skin, which is caused by the electrical activity of the heart. So the way this is done is electrodes are stuck onto the skin, and those detect the electrical activity. and you should get a reading that looks something like this. However, sometimes you wouldn't get a reading that looks like that, and here are the 4 key abnormal heart rhythms that you need to be aware of. Tachycardia first of all, and that is when the heart is beating over 100 beats per minute. Now that is normal if you're exercising, but if you are at rest, that is abnormally high. Bradycardia is when the heart is beating at less than 60 beats per minute. Now, many athletes do have bradycardia as they are so fit that their cardiac muscle can contract harder and therefore fewer contractions are required. But if the heart rate drops too low, an artificial pacemaker would be needed to help to regulate the heart rate.
Fibrillation is when there is either an irregular heartbeat or a chaotic rhythm of the heart. And then finally, we've got the ectopic heartbeat. This is when there are additional heartbeats that are not in rhythm. This will show up on an ECG as 2 heartbeats close together, followed by a normally spaced heartbeat. It is very common for this to occur once a day, but if it's happening more regularly, it could indicate a serious health condition.
We then move on to hemoglobin, and hemoglobins are a group of globular proteins that lots of different organisms have. And this protein is a quaternary structure.
And to clarify, it's a quaternary structure because it has more than 1 polypeptide chains. It just so happens to be that it has 4.
And it's responsible for transporting oxygen. So an oxyhemoglobin dissociation curve is a way for us to view the percentage saturation of hemoglobin with oxygen. And that's normally shown against different partial pressures of oxygen. So what this graph shows us is, at high partial pressures of oxygen, the hemoglobin is at about 100% saturation, so it is fully loaded with oxygen. However, at low partial pressures of oxygen, we have much lower percentage saturations, and that indicates that oxygen is being unloaded. And this would typically be representing different parts of the body. So you'd have a low partial pressure of oxygen in respiring cells or respiring tissues because they're using up that oxygen. So it is an advantage then that the hemoglobin unloads that oxygen and therefore it becomes less saturated because the oxygen can be used in respiration. And in the alveoli, where there's a really high partial pressure of oxygen, you have a high saturation. So where there's lots of oxygen available, the hemoglobin will load on lots to then transport it around. So this graph is showing us changes in affinity, and affinity is referring to the attraction. So at high partial pressures of oxygen, hemoglobin has a high affinity for oxygen. At low partial pressures of oxygen, hemoglobin has a lower affinity. This also links to the concept of cooperative binding. And this cooperative nature of oxygen binding to hemoglobin is due to the hemoglobin changing shape. So we can see here that when first oxygen must be binding, it must be harder because we have got a less steep curve on the graph. But when that first oxygen binds, it actually causes the shape of that protein, the hemoglobin, to change, and it exposes the further binding sites of oxygen more. And that's why the curve is much steeper at this point. It's easier for the second and the third oxygens to bind. It then starts to level off because we now only have 1 binding site of the 4 remaining. So again, that reduces the likelihood of it binding because you've only got a 1 in 4 chance of colliding at the right position. The Bohr effect is when high carbon dioxide concentration causes the oxyhemoglobin curve to shift to the right. So we've got here pH 7.6. But then if we decrease the pH to 7.4 or decrease it further to 7.2, each time that curve has shifted to the right. And this indicates that the affinity that hemoglobin has for oxygen is decreasing. And we can tell that because if we have a look at the same partial pressure, but compare the 2 curves, for the top one, at a low partial pressure of carbon dioxide, which is indicated by the pH, the curve is further to the left, we have a higher affinity and therefore more oxygen is loaded. But at that same partial pressure of oxygen, but with a lower pH, which would indicate there must be more carbon dioxide present because that is what affects the pH, the curve has shifted to the right. And that means that even at the same partial pressure of oxygen, far less oxygen is bound. So it must be unloading more oxygen. And this is an advantage because if you have high levels of carbon dioxide, it indicates it's the site of respiration. So if you're unloading oxygen at that location, there will be a constant supply of oxygen for aerobic respiration to continue.
One quick edit to note here based on marked scheme specificity is for OCR you should be using the terminology associating rather than loading and dissociating rather than unloading.
You might also be asked to compare the haemoglobins of different organisms, starting with a human fetus. And these different labels that we have are, first of all, HbA means adult haemoglobin, HbF means fetal haemoglobin, and then we've got myoglobin, which is a different type of protein altogether. So we're just going to focus on the blue and the green line. Fetal haemoglobin, which is the haemoglobin that a fetus has, the curve has shifted to the left. And that tells us that even at the same partial pressure of oxygen, it is more saturated with oxygen, the haemoglobin is. So it must have a higher affinity. And that's beneficial because the fetus has to be able to get oxygen from the mother's haemoglobin. So it's an advantage that the fetus's haemoglobin has a higher affinity than the adult haemoglobin because it's able to then remove oxygen from the haemoglobin of an adult and it will bind to the fetus's haemoglobin instead. Llamas live at very high altitudes, so they also have a higher affinity. So their haemoglobin has a higher affinity for oxygen. So even though they are in environments with a lower partial pressure, their haemoglobin can still load up oxygen and become saturated.
Next then, it's looking at how carbon dioxide is transported, and this is done in 3 key ways. It can be dissolved and transported in the blood plasma. It could be as carbaminohemoglobin, which is when carbon dioxide can bind with hemoglobin to form carbaminohemoglobin. And then lastly, it could be in the cytoplasm of red blood cells in the form of hydrogen carbonate ions. So a little bit more then about the hydrogen carbonate ions. Almost 85% of the carbon dioxide is transported as hydrogen carbonate ions in red blood cells. Water and carbon dioxide react, and this is a reversible reaction to form carbonic acid. Carbonic anhydrase, which is an enzyme in the cytoplasm of red blood cells, is responsible for catalysing that reaction. Carbonic acid then dissociates to form hydrogen ions and hydrogen carbonate ions. So the haemoglobin can then bind to the hydrogen ions and it dissociates from the oxygen, forming the hemoglobinic acid. The hydrogen carbonate ions diffuse out of the red blood cells and in exchange, chloride ions diffuse into the red blood cells. Both of these ions are negative, so this exchange maintains the electrical balance of the red blood cells and it's known as the chloride shift.
3.13 Transport in plants. So the main substances transported en masse in plants are water and organic substances, meaning anything containing carbon, which is going to be the products of photosynthesis. These are transported either by the xylem or the phloem, which collectively are described as the vascular bundle.
Next time we look at the structure of the roots, and starting with the organisation of the vascular bundles in the roots, because this is actually different to that of the stem and the leaves. In the roots, the xylem is found at the centre of the root, often resembling this star shape with between 3 and 6 spokes, which are the points sticking out. The phloem is found in between each of those star shapes. In the stem, there are several vascular bundles, each containing both xylem and phloem tissues. The xylem is found on the inner edge of each bundle, closest to the centre of the stem, whereas the phloem is found on the outer edge of each bundle, closest to the surface of the stem. In between the xylem and the phloem is a layer of cambium, and cambium is meristematic tissue containing actively dividing pluripotent stem cells. The structure of the leaf, we have the vascular bundle running down the centre of the leaf as a vein, and it contains both the xylem and the phloem tissue. The xylem is towards the top of the leaf, and the phloem is towards the bottom of the leaf within that vascular bundle.
So we're going to start by looking at the structure of the xylem and the phloem.
The phloem tissue is made up of two key cells. We have the sieve tube elements, and then next to those are the companion cells. The sieve tube elements are living cells, but they contain no nucleus, very few organelles, and they have got these perforated end walls. And this is to assist in the mass transport of fluids. Because they are lacking lots of organelles though, they are dependent on the companion cells, which are next to them, to provide the ATP required for active transport of the organic substances into that sieve tube element. Xylem cells, on the other hand, are dead cells and they are also hollow. And here we're looking at a cross section through a xylem in a very thin slice under the microscope. They do not contain any organelles or end walls, and as a result they stack up on each other to create a continuous hollow column, much like a hose pipe, ideal for transporting water and mineral ions. The xylem wall is also strengthened with a waterproof chemical called lignin. So if we have a look at the transport of water, the first thing is knowing the transport of water into the plant. Now this happens from the soil into the root hair cells, and the water is absorbed by osmosis. Root hair cells are adapted to maximise absorption by osmosis by having very thin walls to reduce that diffusion pathway, and also they have those long protrusions providing a large surface area. Once that water is then inside of the root hair cells, it needs to move from that root hair cell to the xylem to be moved en masse. And that can either be through the symplast or apoplast pathway. The symplast pathway is when the water moves through the cytoplasm of a cell. The water will move from cell to cell towards the xylem by osmosis through the cytoplasm, and also through gaps in each cell wall, which is called a plasmodesmata. Each successive cell cytoplasm has a lower water potential, and this is why the water is able to continually move from cell to cell towards the xylem by osmosis. The apoplast pathway is when the water moves through the cell walls. Water can enter the cell wall and move due to cohesive forces. The water molecules stick together because of the hydrogen bonding, and they form a continuous stream of water, which move towards the xylem. This pathway transports water much faster as there is little resistance to the water in the cell wall. The adaptations that some plants have to either very, very high or low quantities of water are also required. So in plants, gas exchange happens through the stomata, and stomata are tiny pores mainly on the lower sides of the leaves. And these can open and close, which is determined by guard cells. And this opening and closing is a mechanism to try and prevent excessive water loss by evaporation. Xerophytes are extremophiles which have adaptations to reduce water loss and are therefore found in environments with limited water, for example the desert. Or marram grass, which is also an example of a xerophyte, is found on the sand dunes, and despite it being next to the ocean, there is still limited water due to the sand being so porous. So if we have a look at some of the adaptations that marram grass, an example of a xerophyte, has. First of all, we've got these curled leaves, and that traps moisture, and therefore it increases the humidity within this space. Because there's an increased humidity, that decreases the water potential gradient and it reduces evaporation. So we can see that we've got not only the curls, but there are also these hair-like structures which would also trap that moist air and increase the humidity. The stomata are sunken in, and that again increases the local humidity to reduce the water potential gradient. On the outside, there is also a thicker cuticle to reduce the water loss by evaporation. In addition to this, to help gain more water, it will have a longer root network so that there is more reach to be able to absorb more water by osmosis. Hydrophytes are the opposites. These are plants which live in or on water, so they require adaptations to survive in an excess of water. Water lilies are an example of hydrophytes which grow on the surface of water. The adaptations include short roots, very thin to no waxy cuticle, and stomata being permanently open and on the top surface of the leaf. These adaptations assure that no additional water is retained in the plant and efficient water loss occurs. Adaptations to ensure enough light is still absorbed for photosynthesis include the leaves being really large and wide on the surface of the water. So next then we're looking at what is transpiration. And this is when water vapour
is lost from a leaf via the stomata. So it's the evaporation of water vapour from the stomata. And you need to know about the different conditions that will affect the rate of transpiration. We're going to have a look at those different conditions, but be aware as well that to measure these changes in rate, you would need to use a piece of apparatus called a potometer. So here are 4 key factors that affect the rate of transpiration. With a higher light intensity, the light will cause the stomata to open more, therefore there's a larger surface area for evaporation. With a higher temperature, the water molecules will gain kinetic energy, move faster, and therefore there'd be increased evaporation. There is a negative correlation between the effect of humidity and the rate. The more water vapour in the air, so the more humid the air, that will make the water potential more positive outside of the leaf, and it therefore reduces the water potential gradient and transpiration. There's a positive correlation with wind, meaning the more windy it is, it will blow away the humid air containing that water vapour, and that will help to maintain the water potential gradient and increase transpiration. This leads into looking at the water of movement up the xylem. So water moves up a plant from the roots against gravity, and in the case of trees that could be several meters against gravity. And this is only possible because of the cohesion tension theory. And this is split into a few different concepts to be aware of.
Cohesion of water molecules, adhesion of water molecules to the xylem wall, and root pressure.
So if we start with cohesion, this links back to the properties of water being polar or dipolar, meaning that we've got these 2 different charged regions. And because of those different charged regions, hydrogen bonds can form between the hydrogen and the oxygen of different water molecules. And that's what these dashed lines are showing. Those water molecules sticking together or cohesing is what creates this continuous column of water in the xylem. Adhesion of water is when water is sticking to other molecules. So water can adhere to the walls of the xylem, for example. Now it's not actually shown in the middle here, but you would have a continuous column of water attached to each other, but also to the walls of the xylem. In this image, I'm just emphasising that it does attach to the xylem walls to really show this impact. And the narrower the xylem, that would mean the larger surface area in contact with the water and therefore stronger adhesion. So we'll actually see this happen in our cohesion tension theory, but as transpiration increases, it can actually cause the xylem to become narrower, and as it gets narrower, that increases adhesion. So we'll come back to that concept.
Quick little change just to make you aware of here for the 2025 version. You only have to say adhesion, not capillarity, and they're really specific on the mark schemes that you say water molecules adhere to the lignin in the xylem walls.
The final thing to consider is root pressure, and this is as water moves into the roots by osmosis, you're increasing the volume of liquid in that space, and that will increase the pressure. Because the pressure is quite high in the roots, it pushes up all of the water above it, so we have this upwards or positive pressure. So we need to look at how these 3 concepts link together to result in cohesion tension theory, which is what causes water to move up the xylem. So the first thing that happens is water will evaporate out of the stomata of the leaves, and as water is being removed, there'll be a lower volume of liquid in the leaves, and that causes the pressure to decrease. Because the pressure decreases, water will move in to fill its space. So more water is then pulled up the xylem to replace it because of that negative pressure. Now this water is all stuck together due to the hydrogen bonds, so they are cohesive. And we have this continuous column of water being pulled up the xylem, replacing the water as it transpires. But because the water molecules are also stuck or adhering to the walls of the xylem, as the water column is being pulled up and the water is stuck to the walls of the xylem, it pulls the water column upwards and the walls of the xylem inwards, and that makes the walls of the xylem closer together, and therefore the lumen is narrower. So it's this tension of the water being pulled upwards in that continuous column that pulls the xylem to be narrower. Now because it's narrower, that adhesion will have a bigger impact, And we've also got the root pressure pushing the water up, and those three properties together are what ensure we get this continuous column of water moving up the xylem against gravity. The second type of transport is the transport of the organic substances produced in photosynthesis. So that is what we're going to be looking at, how those organic substances are transported around the plant through the phloem. So we've already had a look at the structure of the phloem, and this then leads us into translocation. And this is the transport of organic substances in plants. And this process requires energy. It's an active process. It revolves around this idea that we have the mass flow from the source, and the source is where the organic substance is made, so in photosynthesizing cells. And we have the mass flow from that source to the sink, and the sink is the site where the organic substances are going to be used in respiration.
A few little mark scheme specific additions / changes here to be aware of that I've added in for this 2025 version are, first of all, although glucose is what is made in photosynthesis, it is sucrose that is transported, and you do have to say sucrose. It's also amino acids, and those are used up in respiring tissues. The sucrose and the amino acids can be used as well. And instead of just saying taken up, you'd need to know they are assimilated.
So you can look at this model here, the source to sink explanation. It's a very simplified version, but it demonstrates what is happening. We've got our source cell, which we said is the photosynthesizing cell, and that will be connected via the phloem to a sink cell, which is a respiring cell. And we have the xylem next to the phloem in the vascular bundle. But to help represent this, we're just saying it's in a tank of water. So when that cell is photosynthesizing, we're going to be producing sucrose, which lowers the water potential of the cell. Therefore, water would move from the xylem into that cell. At the other end, the respiring cell is using up sucrose, and therefore it has a more positive water potential compared to the surroundings, and water would leave the sink cell by osmosis. So what that does to the hydrostatic pressure is, at the source cell where water is moving in, the hydrostatic pressure will increase, and at the sink cell where water is moving out, the hydrostatic pressure would decrease. So as a result, the source cell has a comparatively higher hydrostatic pressure than the sink cell, so the liquids within those cells are forced through the phloem towards the sink cell. Now, looking at a bit more detail of what is actually going on takes us into the next stages of translocation. So we've already said that the photosynthesizing cell is the source cell, and at that point organic substances are being created by photosynthesis, for example sucrose, and that's what's shown in this image in the red.
So let's have a look at how sucrose transports from the source to the sieve tube element. First of all, this is an example of active transport because it's hydrogen ions being actively transported, and this occurs from the companion cell into the photosynthesizing cells of the source. This creates a concentration gradient, and therefore the hydrogen ions move down their concentration gradient via carrier proteins back into the companion cells. Lastly, then we have the co-transport of sucrose with the hydrogen ions occurring via protein co-transporters, and that transports the sucrose into the companion cells. The sucrose then diffuses through plasmodesmata into the sieve tube elements.
Once it's in the sieve tube element, that is now going to be decreasing the water potential. And as a result, water from the xylem will move into the phloem by osmosis. That will increase the hydrostatic pressure at this end of the sieve tube element. At the same time, sucrose is being used in respiration at the sink, or it might be stored as insoluble starch. That means that more sucrose is going to be actively transported into the sink cell, which causes the water potential to decrease at this end. And as a result, water will move from the sieve tube element back into the xylem. So this means that at this end of the sieve tube element, we have a much lower hydrostatic pressure. So as a result, we've got a high pressure, a low pressure, we'll have the movement of solutes down that pressure gradient, and that is what causes the solution to move through the phloem. And that takes us to the end of module 3.
So we begin with homeostasis then, which is the maintenance of a constant internal environment via physiological control systems. And these control systems keep body temperature, blood pH, blood glucose and blood water potential within the set limits. And this is because homeostasis involves negative feedback loops.
So a negative feedback loop is the most common one that we see. And this is when a deviation from the set limits is detected in the body, and that'll be detected by a receptor, and then mechanisms are put in place to bring those conditions back within these set limits. So we can see here an example of that with regulating the body temperature. It involves the nerve system, and also it often involves hormones as well. In contrast, positive feedback is actually quite rare, and this is when a deviation from the set limits triggers a response to increase the deviation from that set limit even further. So a good example to demonstrate this is during childbirth. And when the baby's head presses on the cervix, it causes the hormone oxytocin to be released. And that release and deviation from the set limit of that hormone causes the uterus to contract, but it results in even more oxytocin being released, so even more contractions in the uterus. So if we start now with an example, which is thermoregulation. And the reason that we have to maintain the temperature in our body is if the temperature was too low, there wouldn't be enough kinetic energy for enzyme-controlled reactions. And if the temperature was too hot, enzymes would denature and then reactions would also stop. So it's really important either way so that metabolic reactions continue at a fast enough rate so that cells don't die.
So exotherms control their internal temperature less well than endotherms, and they do so mainly by changing their behaviour. They do have some physiological responses too though. And in fact, most animals are exotherms, for example fish, amphibians, reptiles and invertebrates. Exotherms within aquatic environments wouldn't have much need to regulate their body temperature, and this links back to what you learn about water having a high specific heat capacity, and therefore it buffers temperature changes and the temperature of water remains relatively constant. Exotherms on land have a bigger challenge as the temperature of air fluctuates far more. So exotherms have a range of behavioural responses to help regulate their temperature. To keep their body temperature high enough to ensure that metabolic reactions can occur fast enough, they have to warm up through conduction against hot surfaces. So for example, we can see here basking on warm ground. And to cool down, they would have to move to the shade or move into water, dig underground or lie on colder surfaces.
Endotherms, in contrast, do regulate their internal body temperature using the nervous system. And this is done through a whole range of mechanisms. If we start by looking at the receptors, the peripheral receptors are in the skin, and that detects a change in the external temperature. That would then send an impulse along the sensory neuron to the brain, where the hypothalamus coordinates that impulse. And that would trigger a range of responses linked to either glands or muscles. So for example, the sweat glands, if you were hot, would produce more sweat, and that would provide a cooling impact. Vasodilation and vasoconstriction are both linked to muscles, and it's to do with the arterioles in the skin either having more contraction or less contraction to result in the dilation or the constriction of those blood vessels. And that controls the amount of blood flow to the surface of the skin, and that will then control the amount of heat energy that can radiate from that blood close to the skin surface. So if you were hot, then you would undergo vasodilation to increase the amount of heat radiating. If you were cold, you would go through vasoconstriction, and that results in less heat energy radiating from the skin. Shivering is also an example of muscle response, and it's the contracting and the relaxing of skeletal muscle. And that will increase the rate of respiration, and therefore more heat energy would be released. Now, animals that have a lot of fur or feathers can also raise their fur or feathers upwards, and that will then trap a layer of air to insulate. Or if they're really hot, they would then lower the fur and hair so that less air is being trapped to prevent less of an insulating layer. And the muscles that are involved in this response are the erector pili muscles within the skin. The last option here is to do with modifications in behaviour. And we see that in humans, for example, moving to the shade if you're hot, or taking off clothes if you're hot, using a fan. But you also see it in other animals. So other animals will also move to the shade if they're hot, and penguins huddle when they're cold. So we now move on to 5.1.2, excretion as an example of homeostatic control. Metabolic reactions, which are continuously happen, create waste products, which can become toxic if they are not removed. And the removal of this waste product is known as excretion. The two key examples that you need to be familiar with are carbon dioxide and nitrogenous waste. Carbon dioxide is the waste product from respiration, and it's excreted from the lungs when you exhale. Nitrogenous waste, for example urea, is created from excess amino acids in the diet. And unlike glucose, excess amino acids can't actually be stored and used at a later date. And that means amino acids are broken down, and that happens in the liver. And they're broken down to ammonia, which is toxic, and then urea. That urea, if it's in a high enough concentration, would also be toxic. So it gets excreted through the kidneys. Now, mammals produce urea as nitrogenous waste, but fish produce ammonia and birds produce uric acid. So we said that the liver has a role in that formation of urea, but it actually also is involved in glycogen, where we have the glucose being stored away as glycogen, and the breakdown of other toxins, so detoxification. And in order to do this, the liver contains a range of enzymes. It's a very large organ, and it receives oxygenated blood through the hepatic artery, and the blood leaves the liver through the hepatic vein. The hepatic portal vein also supplies the liver with blood, and that is supplying blood from the digestive system. So you need to be not only aware of the functions, but the structure and the histology of the liver. So liver cells are called hepatocytes. They have many mitochondria, large nuclei, and prominent Golgi apparatus. And these adaptations enable a really high metabolic rate. So here we're going to look at the structure and the histology of the liver. So we can see the liver here, and that is made up of these functional units called lobules. And that is all of these hexagon shapes that we can see. And this is then zooming in on one of those lobules. So a lobule is essentially like the equivalent of the nephron in the kidney. It's where all the action is happening. And we can see then on this lobule that the blood is delivered to the lobule through the hepatic portal vein and the hepatic artery. And that's what we've got here, the portal veins, which is bringing the blood from the digestive system, rich in everything that's just been absorbed from digestion. And then we've got the hepatic arteries as well, shown in red, bringing the blood as well. And then the blood that's been delivered from the hepatic artery and the hepatic portal vein is then going to mix in these vessels that we can see here, which are essentially spaces that are surrounding the hepatocytes, which are the cells of the liver. And that is where we have blood from the hepatic arteries and the hepatic portal veins mixing. So we actually have oxygenated and deoxygenated blood mixing at that point. Now it's important because the blood delivered from the hepatic artery is really highly oxygenated, and this oxygen can mix with the blood from the hepatic portal vein, so therefore we have at least partially oxygenated blood at this point. Now that is going to run through those sinusoids, past all of these hepatocytes, which we're going to look at their role shortly, and then it all drains into the hepatic vein. So all of that blood eventually drains in the hepatic vein, and then it gets transferred away. Now a little bit more then about the structure. This is showing you an entire lobule, and we're going to zoom in now on just like one segment within that lobule. And here's just one segment, and this is the outside where we have the hepatic artery, and in this case it's an arteriole. We have the hepatic portal vein leading to a venule. Now the first thing is you need to be able to identify these, and the way you can actually do that is by looking at the thickness of the wall. The hepatic arteriole will have a thicker wall compared to the hepatic portal venule. And as we said, those will be delivering deoxygenated blood from the hepatic portal venule, oxygenated blood from the hepatic arteriole, and that mixes in those spaces in the middle called the sinusoid, and that all eventually drains toward the hepatic vein. We do have a final section here shown in green, and this is the bile duct. And the way we can tell it's a bile duct and not one of the blood vessels is it doesn't have a wall, instead it is surrounded by hepatocyte cells. So let's have a look at some of this information then. To protect against disease, within those sinusoids, there are Kupffer cells. And those Kupffer cells are able to help destroy any pathogens that might enter. So they're a bit like macrophages and engulf those foreign particles. The hepatocytes, which are the cells that make up the liver, which are shown in grey in this image, they produce bile, and they'll be using the products from breaking down old blood and blood cells to do that. The bile is first secreted from those hepatocytes into spaces called canaliculi, for plural, or for singular, a canaliculus. And we've just got one here, so this is our canaliculus. And the bile is produced in the hepatocytes, secreted into this canaliculus, and then it drains and moves down into that bile duct. And from the bile duct, it will then go to the gallbladder where it is stored.
Just a quick interruption here with a 2025 edit on this bullet point here, where you can see I've added in more details. So you also need to know that they are macrophages, engulfing any pathogens that may have entered through the blood delivered by the hepatic portal vein, which came from the digestive system.
So some other functions then to talk about. The hepatocytes, which were those liver cells, in response to insulin, they can also absorb excess glucose from the blood and convert it to glycogen. In response to glucagon, the hepatocytes will hydrolyze glycogen back into glucose and release it into the blood. Detoxification also happens here, and this is the neutralization and breakdown of unwanted chemicals such as alcohol, drugs, hormones, and toxins produced in metabolism. As we've already said, many metabolic reactions produce toxins, and the liver contains enzymes that can break those down into non-toxic substances. Finally, the ornithine cycle, which is the urea cycle, is also occurring here. And this is how specifically the waste substance urea is produced from ammonia, which we'd already mentioned is what excess amino acids are broken down into. And this will then be ready to transport that urea to the kidneys for excretion. So excess proteins from our diet, as we said earlier, cannot be stored, and instead they go to the liver to be deaminated. And deamination is when the amine group is removed from the amino acids, and that is what converts it into ammonia. Ammonia is highly toxic, which is why it has to be converted to urea before it's then transported in the blood to be released. Urea is also toxic, but only in very high concentrations, which it shouldn't be in, because when the blood gets to the kidney, the urea is filtered out. Now that leads us into the kidney. This is responsible for the excretion of that urea, the nitrogenous waste, and also osmoregulation, which is the process of controlling the water potential of the blood. In this case, it's the renal artery that supplies the kidney with blood to be filtered. And we can see that here shown in red. There's our renal artery entering. And the renal vein is going to be taking that filtered blood away. Kidneys are made up of 3 distinct layers. We have the cortex, the medulla, and the pelvis. The cortex is the dark outer layer that contains many capillary networks carrying blood from the renal arteries to the nephrons. The medulla is this section here, and that contains the nephrons. The pelvis, which is shown here in yellow, is where the urine collects before leaving the kidney and travelling to the ureter. So if we focus then on the structure of the nephron, you have 2 kidneys, and we just looked at the structure of those kidneys. And within the medulla, you have millions of nephrons. And the nephrons are the structures within the kidney where the blood is filtered, and then useful substances are reabsorbed back into the blood. The blood is filtered here to remove waste, but to selectively reabsorb useful substances back into the blood. So in terms of the structure, we have the Bowman's capsule, which is on the outside of the glomerulus, and that is where ultrafiltration occurs. That then leads into the proximal convoluted tubule. And we're going to look at how glucose is reabsorbed after it's been filtered at that point. We then have that leading into the loop of Henle. And here sodium ions are actively transported out of the ascending limb, which is the one going up, into the medulla to create a lower water potential. And then water is then going to be moving out by osmosis from the descending limb of the loop of Henle, as well as it will continue to move out from the distal convoluted tubule and also the collecting ducts. And that's all due to water potential gradients. So eventually any remaining liquid in the collecting duct, that goes on to form the urine, and that liquid will contain water, dissolved salts, urea, and other substances such as hormones. So in a little bit more detail, we said that the ultrafiltration is happening in that Bowman's capsule, or the renal capsule. And that is because within that capsule, there are lots of blood vessels, which we call the glomerulus. And blood enters through an afferent arteriole, and that splits into lots of smaller capillaries, making up that glomerulus. Because you are going from a wider diameter arteriole into smaller diameter capillaries, that creates a high hydrostatic pressure of the blood. And water and small molecules are then forced out of the tiny gaps between the capillary walls, and that forms the glomerulus filtrate. Once the fluid is forced out of those capillary walls, it then passes through a basement membrane. The first image is showing you the capillaries. We've got the blood and the red blood cells flowing through those capillaries, and there's these tiny gaps in the capillary walls that the small molecules and water are forced out of. Then after the capillaries, as we can see in this image, there is another layer which is the basement membrane, and that's made up of a network of collagen fibers and proteins. And the fluid then has to pass through that basement membrane, and that acts like a sieve. Now, the Bowman's capsule wall also has podocytes, which are special cells that act as an additional filter. So that's what we can see here, these podocyte cells that surround that basement membrane. So large protein and blood cells are too big to fit through the gaps in the capillary endothelium. They remain in the blood, and this blood then leaves via the efferent arteriole. After that glomerulus filtrate has been formed in that Bowman's capsule, it enters the proximal convoluted tubule. And at this point, almost all of the filtrate is reabsorbed back into the blood, leaving just urea, excess mineral ions, and water behind.
Adding this into the 2025 edition, not a change in the spec, just extra detail to make sure it's really marked scheme specific. You do also have water being, or some water being reabsorbed at this point due to the changes in the water potential. So that transport of glucose has resulted in approximately 80% of the water in the PCT also moving back into the capillaries at this stage because of that water potential gradient.
Now, one of the key things that's filtered out that gets reabsorbed is glucose and small amino acids as well, but we're just going to focus on the glucose in particular. And the way this gets reabsorbed in the proximal convoluted tubule is almost identical to how it is absorbed in the small intestines. Is co-transport. So the concentration of sodium ions within these epithelial cells of the proximal convoluted tubule cell is decreased as sodium ions are actively transported out of that cell and into the blood capillaries. Now, due to the concentration gradient of the sodium ions in that PCT cell compared to the lumen of the PCT, that results in the sodium ions being able to move down their concentration gradient into the cell by facilitated diffusion. But this happens through a co-transporter protein, and glucose is co-transported in with those sodium ions. And in this way, any of the glucose that was forced out in ultrafiltration that is now in this glomerular filtrate in the lumen of the proximal convoluted tubule, it gets reabsorbed into the proximal convoluted tubule cell, and then by facilitated diffusion it will enter the bloodstream. And in that way, almost all of the glucose is reabsorbed from the filtrate back into the blood. And that's important because glucose is not a waste product, it is needed for respiration, and if there's too much of it, it can be stored in the liver as glycogen. So now that filtrate will enter the loop of Henle. And the loop of Henle plays a key role in maintaining a sodium ion gradient and therefore water being reabsorbed. So the filtrate passes into the loop of Henle, and it's going to enter first of all the descending limb, which in this image is shown on the right hand side. Sometimes you might have an image where you don't have this overlap in the tubes, so therefore it appears on the left hand side. But the key thing is it's connected to the PCT and the tube goes down first, so that is the descending loop of Henle. That then leads into the ascending limb of the loop of Henle. And mitochondria in the walls of the cells provide energy to actively transport sodium ions out of the ascending limb of the loop of Henle. So lots of sodium ions are being actively transported out, and as a result, this space in the medulla, there'll be an accumulation of sodium ions, and that lowers the water potential of the medulla compared to the water potential in the filtrate. So that then means that as more of that filtrate is passing down the descending limb, the water from that filtrate is going to diffuse out by osmosis into this interstitial space in the medulla. And that water, once it's in the medulla, will then be reabsorbed back into the capillaries. At the base of the ascending limb, some of the sodium ions are transported out by diffusion, as there's now a very dilute solution, as the solution surrounding the loop of Henle is now quite dilute because of all the water that has moved out by osmosis.
A 2025 edit here, just making it really clear that you're actually maintaining a sodium and chloride ion gradient, not just a sodium ion gradient. So the information we just went through, exactly the same, except you have sodium and chloride ion gradient enabling the absorption, and you get the accumulation of sodium and chloride ions outside the nephron in the medulla, which is what lowers the water potential.
So that filtrate now will enter the distal convoluted tubule. And due to all of those sodium ions that have been actively transported out of the loop of Henle, when the filtrate reaches the top of that loop of Henle, it's very, very dilute when it enters the distal convoluted tubule. So that filtrate moves into the distal convoluted tubule, and this section of the medulla is actually very concentrated. So there is a very low water potential on the outside of the distal convoluted tubule. And therefore, even more water is going to diffuse out of the distal convoluted tubule and out of the collecting duct. That water will then be reabsorbed into the blood.
Another 2025 edit here, same concept as it was on the previous slide. You've got to say due to the sodium and chloride ions, not just sodium ions here.
So anything that remains in the collecting duct then goes on to form urine. So that is how the kidneys filter the blood, but the kidneys also have a role in regulating the water potential of the blood. And this involves negative feedback and also the brain. The hypothalamus within the brain, which we can see here, detects changes in water potential. And that is because of receptors that are found in this location called osmoreceptors. If the water potential of the blood is too low, water moves out of those osmoreceptors by osmosis and it causes them to shrivel. And that stimulates the hypothalamus to produce more of the hormone ADH. So the hypothalamus constantly produces ADH, but it will produce more of that hormone if the water potential of the blood is low. If the water potential of the blood is too high, water will enter those osmoreceptors by osmosis, and that stimulates the hypothalamus to produce less ADH. So the hypothalamus is where the hormone is produced, but that hormone is then transported to the posterior pituitary gland, and it's from there that it is released into the blood. The ADH will then travel through the blood to its target organ, which is the kidney. ADH, or antidiuretic hormone, will bind to complementary receptors that are only located in the kidney, specifically on the cells in the distal convoluted tubule and the collecting duct. When it binds, it activates the enzyme adenyl cyclase, and that makes cyclic AMP, or CAMP. This activates an enzyme which causes vesicles containing proteins, known as aquaporins, to fuse with the membrane, and those aquaporins are channel proteins that allow water to be transported from the collecting duct or the distal convoluted tubule back into the blood. So as a result, the membrane becomes more permeable to water and more water will leave to be reabsorbed into the blood. So that's what we can see happening here with those aquaporins. And as we said, that causes more water to leave the nephron from the distal convoluted tubule or from the collecting duct to be reabsorbed back into the blood in the capillaries. And that means that the urine that remains in that collecting duct will be more concentrated and there'll be a lower volume. So if we just quickly summarize then this negative feedback, we've got the water potential of the blood increases, meaning there's too much water in your blood. That's detected by osmoreceptors in the hypothalamus. The hypothalamus will then release less ADH, which is released by the posterior pituitary gland into the blood. That hormone will then bind to the receptors on the cells in the DCT and the collecting duct walls. That means there'll be less of that hormone attaching to the DCT and collecting duct walls, so fewer aquaporins are embedding, so the walls are less permeable to water. That means less water is reabsorbed back into the blood, and instead it remains in the urine, so you have larger volumes of dilute urine, and that brings your water potential back to the normal set limits. In contrast, if you don't have enough water in your blood, that means the water potential of the blood has decreased. That will be detected by osmoreceptors in the hypothalamus and cause an increase in the release of ADH from the hypothalamus, and that will mean more of that hormone is released by the posterior pituitary gland into the blood. That causes the DCT and the collecting duct walls to become more permeable to water. More water is reabsorbed into the blood and less is lost in the urine. And that then brings our water potential back within its normal levels. Now, urine can actually be used for diagnosing a range of things. The fact that urine is composed of substances filtered out of the blood is why it can be used for this. So it can be tested for diabetes, pregnancy, the presence of anabolic steroids, and also other drugs. Pregnancy test uses monoclonal antibodies to detect the presence of the human growth hormone, which is produced by pregnant women. A monoclonal antibody is a single type of antibody that can be isolated and cloned. So in our pregnancy test, which we can see over here, at point A, we have a urine sample, and there is an absorbent piece of paper at the end of the test. So the absorbent end of the pregnancy test is absorbed in the urine. That urine is then absorbed and it moves up the pregnancy test. And at position B of the pregnancy test, we have a mobile monoclonal antibody. And that monoclonal antibody is complementary in shape to the human growth hormone, which you only produce if you're pregnant. So in this case, the person is pregnant and the human growth hormone is in the urine, and it binds to those mobile monoclonal antibodies, which have a coloured dye attached to them. As the urine moves up the paper, those mobile antibodies move with it. And at position C, there is a second monoclonal antibody, but this one is immobilized in this fixed position. It's also complementary to the human growth hormone. So if any of the first antibodies do have it attached, they will bind at this point and stay in that fixed position. And because those have got coloured dye on them, you get a blue strip. Now at the very end at position D, there is a third antibody which is immobilized, and this is complementary in shape to the constant region of the first antibody. So that means any of those antibodies that are moving along will attach, and you get a second blue line. And that is our control strip, just to show that the pregnancy test is working and that those antibodies at position B are moving. So if you have 2 lines, it indicates you're pregnant. 1 line, you are not pregnant. Anabolic steroids are also excreted in urine, and therefore they can be tested for in a similar way to this, along with many other drugs that are excreted in your urine. They could also be tested in a similar way.
Just a quick 2025 edit correcting, there was a typo in this section here. I think it said human growth human, but what it should have been referring to was the hormone HCG. So that is the hormone that is present in the urine of pregnant women that is tested for using these monoclonal antibodies.
Now, sometimes the kidneys do fail, and there are many causes of kidney failure, which could be infection, high blood pressure, genetic conditions, or even physical damage. Kidney infections and high blood pressure can damage the tubules, podocytes, epithelial cells, and the basement membranes of the Bowman's capsule. And as a result, that would affect the filtration, and large molecules would be able to filter out of the blood as well, such as proteins, large proteins, and red blood cells. If the kidney completely fails, then the blood will not be filtered properly, and this can lead to a buildup of urea and blood and an electrolyte imbalance. The glomerular filtrate rate, or GFR, is affected by kidney failure and can be measured as an indication of disease. And this is measured indirectly by testing the blood for creatine levels. And creatine is a breakdown product of muscles. If the levels of creatine in the blood increase, it indicates that the kidneys are not filtering properly. So your options for treatment are 2 types of dialysis. We've got hemodialysis, which is the one which is connecting your body to the machine. and the blood is going to come out of your body and the machine itself filters it. Now, these are quite successful in filtering the blood. However, long-term dialysis can have really harmful side effects, and therefore the best treatment would be a kidney transplant. However, there are a lack of available donors, and there's always the risk of organ rejection. So if you do have a kidney transplant, you would have to be on immunosuppressant drugs for life to reduce the risk of rejection. And because you're on immunosuppressant drugs, you are more at risk of getting ill from infections. The other thing to be aware of is most transplanted kidneys are only going to work for about 10 years. So you might then have to have a second organ transplant. Then we move on to communication involving neurons and the nervous system. So we'll start by looking at the neurons. The nervous system is made up of billions of neurons, and these can be categorized into sensory, relay, and motor neurons. All three have some common features. They all have a cell body which contains the organelles found in a typical animal cell, including the nucleus. They also have proteins and neurotransmitters which are made in the cell body. Dendrons carry the action potential to surrounding cells, and they all have an axon, which is a conductive long fibre that carries the nerve's impulse along the motor neuron. You also need to be aware about myelinated neurons, and a myelinated neuron has a Schwann cell which wraps around the axon to form a myelin sheath. And that's what we can see here. The yellow is representing that myelin sheath. And that is a lipid, and therefore it does not allow charged ions to pass through it. So in that way, it prevents the conduction of the electrical impulse. There are gaps between these myelin sheaths, and these are called the nodes of Ranvier. The action potential therefore has to jump from node to node, and that is known as saltatory conduction. And that means that the action potential travels much faster because it's only being generated at a number of positions instead of at every single distance along the entire axon. Now, knowing the different structures of the three is key as well. So the sensory neurons, these carry electrical impulses from the sensory receptor cell to the relay neurons, and also sometimes to the motor neuron and the brain. They have a long dendron, which carries the impulse from the sensory receptor cell to the cell body, and then an axon to carry the impulse to the next neuron. The relay neurons carry impulses between the sensory and the motor neurons. They have multiple short axons and dendron.
We then have the motor neurons, and these carry impulses from a relay or a sensory neuron to the effector, which will be the muscle or gland, and this is the part of the body that is carrying out the response. They have one long axon and multiple short dendrites.
So the sensory receptor cells are responsible for detecting a stimulus. And these cells are transducers because they'll convert different types of stimuli into electrical nerve impulses. There are different types of sensory receptors, and each of them can detect a different stimulus. So here are three key examples and the stimulus that they can detect. So let's have a look then at the Pacinian corpuscle. These pressure receptors are located deep in the skin, mainly in the fingers and the feet. The sensory neuron in the Pacinian corpuscle has a special channel protein in its plasma membrane. So that's what we're looking at here. The Pacinian corpuscle is essentially a sensory neuron that is wrapped up in lots and lots of layers of lamellae. Now, those membranes of the Pacinian corpuscle that are wrapping around the end of this sensory neuron have stretch-mediated sodium channels. So these are protein channels embedded within the membrane that will open when there is pressure applied and therefore stretching the membrane. And when those channels open, sodium ions will enter, and if enough sodium ions diffuse in, it will generate an action potential. So in that way, when the pressure is applied and it deforms the neural membrane, it stretches and widens open those sodium ion channels to generate an action potential. If there is enough stretch to enable enough sodium ions to enter to reach plus 40 millivolts. When a neuron is not conducting an impulse, there is a difference between the electrical charge inside and outside of the neuron, and this is known as the resting potential. There are more positive ions, so sodium and potassium ions, outside of the neuron compared to inside of the neuron, and therefore the inside of the neuron is comparatively more negative at minus 70 millivolts. So that's what we're seeing on this graph. The potential difference, or the voltage, at this point when there is no stimulus, it remains constant at minus 70, and that is the resting potential. So the way that that resting potential is established and maintained is by the following steps. It's maintained first of all by a sodium-potassium pump, and that's shown here in purple. And this involves active transport and therefore ATP. The pump will move 2 potassium ions into the axon and 3 sodium ions out of the axon or the neuron. This creates an electrochemical gradient causing potassium ions to diffuse out and sodium ions to diffuse in. However, the reason that we end up with a more negative inside compared to outside is the membrane is more permeable to potassium ions. And there's a couple of reasons why. First of all, many of the potassium ion channels are permanently open, whereas the sodium ion channels aren't always open. And the second reason is there are more of these channels embedded in the membrane. So those reasons mean
More potassium ions are going to diffuse out.
So you have a comparatively higher charge on the outside compared to inside, and that's how we get this minus 70 millivolts. Now, an action potential is when the neuron's voltage increases beyond a set threshold, and that then results in the generation of a nervous impulse. An increase in the voltage is known as depolarization, and that is due to the neuron membrane becoming more permeable to sodium ions. And therefore, sodium ions will be entering the neuron, and that is what creates this more positive voltage until eventually it does get to the point where it is at the maximum level of plus 40. And once an action potential is generated, it moves along the axon like a Mexican wave. So essentially following this change in voltage at every position, or in this case at every node of Ranvier, along the axon. We're going to go through then all of these stages of the graph, so this change over time at one position in the axon, and what causes these changes in voltage. So until there is a stimulus, we are at our resting potential. And then at this point, we can see there is a stimulus. And a stimulus provides energy that can cause the voltage-gated ion channels, the sodium ion channels, in the axon membrane to open. And that will cause sodium ions to diffuse in, which increases the positivity of the axon. So here we have what is happening at resting potential. But then when we have that stimulus causing the sodium ion channels to open, we can see that is now open, and we will get more sodium ions diffusing in. This causes even more voltage gated channels to open, so even more sodium ions are going to diffuse in. And that's why the voltage becomes more positive. When a threshold of minus 55 millivolts is reached or exceeded, you will then always reach this maximum of plus 40 millivolts. And when you reach that point, voltage-gated sodium ion channels close, but the potassium ion channels stay permanently open. So that results in no longer having any more sodium ions diffusing in, but you still have potassium ions moving out. And that is why we get this repolarization occurring, which is when the voltage decreases again. We then have a potassium ion channel that opens at this point, so even more potassium ion channels leave. And this is how you get this overshoot, where you get hyperpolarization, meaning that the voltage becomes even more negative than the resting potential. So we can see here it is now in what we call the refractory period, and during that period you wouldn't be able to generate another action potential. So that is how one action potential is generated, and that would have happened just at this first position on the axon. But once an action potential is generated, that can then trigger the voltage-gated sodium ion channels at the next position on the axon to open, and you go through this whole action potential again. And it happens at all of those nodes of Ranvier, and we describe that as being like a Mexican wave. The all or nothing principle is linking to this idea of the threshold. So if depolarization does not exceed the minus 55 millivolt threshold, an action potential and an impulse are not produced. So nothing will actually happen. Any stimulus that does trigger depolarization to the threshold of minus 55 millivolts will always peak at the same maximum. So the bigger the stimulus, you don't get a higher voltage. Instead, it's the frequency of these action potentials occurring, that will increase. And that's what we mean by the all or nothing principle. And it's really important as it makes sure that animals only respond to large enough stimuli rather than responding to every slight change in their environment, which would overwhelm your senses and actually then potentially put you at danger instead of protecting you against it. We saw the refractory period within that action potential, and after an action potential has been generated, the membrane enters that refractory period when it can't be stimulated again. And that's because those sodium channels are recovering and can't be opened. Now, this is important for 3 reasons. Number 1, it ensures that discrete impulses are produced. An action potential cannot be generated immediately after another, and this makes sure that each is separate and you can respond to each one individually. It also ensures that action potentials travel in one direction, and this stops the action potential from spreading out in 2 directions, which would prevent a response from happening. And number 3, it limits the number of impulse transmissions, and that's important to prevent an overreaction to a stimulus. So the action potentials are what travel along the axon of a neuron, but there are gaps between these neurons called synapses. And at this point, the action potential can't jump across, and instead neurotransmitters have to diffuse across this gap, the synaptic cleft, to then generate a new action potential in the next neuron. So let's have a look at this whole process of how it happens. Here is our presynaptic neuron, and as an action potential arrives at the end of that neuron, which is known as the synaptic knob, depolarization of that synaptic knob causes calcium ion channels to open, and therefore calcium ions will diffuse into the synaptic knob. Now that influx of calcium ions causes these vesicles here, which contain neurotransmitter, to move towards the presynaptic membrane. They then fuse with the membrane, and that causes the release of the neurotransmitter into this gap, which is known as the synaptic cleft. Because the neurotransmitter is only released from the presynaptic neuron, we then have a really high concentration on this side of the synaptic cleft compared to the other. And that means the neurotransmitter diffuses across the synaptic cleft down its concentration gradient. And on this postsynaptic neuron, on the membrane, there are receptors that the neurotransmitter binds to because they are complementary in shape. When those neurotransmitters bind to the receptors, it causes sodium ion channels that are embedded in the postsynaptic neuron's membrane to open, and therefore sodium ions will diffuse into the postsynaptic membrane. If enough sodium ions diffuse in to go above that minus 55 millivolt threshold, then depolarization occurs and a new action potential is generated in the next neuron. Now that neurotransmitter doesn't remain permanently bound, and if it did, it'd be constantly generating a new action potential even if there wasn't a stimulus. So for that reason, there are enzymes within the synaptic cleft that are able to break down the neurotransmitter, causing it to be released, and the neurotransmitter then gets reabsorbed and recycled. So this is unidirectional. The neurotransmitter is only released from the presynaptic neuron, therefore it moves down its concentration gradient, and there are only receptors on the postsynaptic neuron. So that is what makes sure it's unidirectional. Now, an example of this is a cholinergic synapse. It's exactly that process that we went through, but you just need to be aware of the fact that the neurotransmitter is called acetylcholine, and the enzyme that breaks down the acetylcholine after it's bound to the receptor is acetylcholine esterase. And that will hydrolyze the acetylcholine into choline and acetate. Those get reabsorbed and can be reused again. Now, we did say that it's only going to generate an action potential in the postsynaptic neuron if you reach the minus 55 threshold. And to make sure that happens, summation is involved. And summation is this rapid buildup of neurotransmitter in the synapse to help generate that action potential. And there's 2 types. We have spatial and temporal. Spatial summation is when you have multiple different presynaptic neurons all converging at one postsynaptic neuron. And collectively, they'll release enough neurotransmitter to bind to enough receptors to generate an action potential in the postsynaptic neuron. Temporal summation is when you just have one presynaptic neuron next to one postsynaptic neuron. And the repeated stimulation of that presynaptic neuron will over time add up to enough neurotransmitter to exceed the threshold. Some synapses are inhibitory though. So an inhibitory synapse causes chloride ions to enter instead of the sodium ions. And it can also cause potassium ions to move out. This makes the membrane potential even more negative, so it can be at about minus 80 millivolts. So your postsynaptic neuron will go into hyperpolarization, and therefore it's really unlikely that an action potential would occur because you would need even more sodium ions to move in to reach that minus 55 millivolts. We next move on to hormonal communication. And for this, we need to look at the endocrine system. And this is made up of endocrine glands and it's responsible for the hormonal communication. Endocrine glands secrete hormones and those are then transported in the blood where they will then bind to receptors on the target cells of the target organs. And we can see here a selection of the different organs that make up the endocrine system. So a hormone is a chemical messenger transported in the blood. Hormones have widespread and longer lasting effects compared to the nervous system response. And hormones could be steroids, proteins, glycoproteins, polypeptides, amines, and tyrosine derivatives. Steroid hormones are lipid soluble and they can diffuse across the cell surface membrane into their target cell to bind to a receptor often located within the cytoplasm. So for example, estrogen. Non-steroid hormones are insoluble in lipids and therefore cannot diffuse across the cell surface membrane. Instead, they bind to complementary shaped receptors on the surface membrane of the target cell, for example, insulin. This binding to a receptor causes a cascade of responses within the cell, and that is how the hormone causes a response. So if we have a look at the adrenal glands as an example of an endocrine gland, humans have two, and they're located on the top of each kidney. Adrenal glands are made up of adrenal cortex and the adrenal medulla, and that is then surrounded by a capsule, which is the outer layer. The adrenal cortex and the adrenal medulla sections both secrete hormones. So let's have a look at these two sections. The adrenal cortex is controlled by hormones secreted by the pituitary gland, located in the brain. There are three types of hormones that the adrenal cortex can secrete, which are these three just listed here. The adrenal medulla is controlled by the nervous system. When the sympathetic nervous system is stimulated, it causes the release of adrenaline, which can increase heart rate and raise blood glucose concentration, and noradrenaline, which increases heart rate, causes pupils to dilate, widens the airways in the lungs, and narrows the blood vessels in non-essential organs to create a higher blood pressure.
A 2025 edit here, and that is just making you aware of the roles of these three hormones as well. So we've got cortisol, for example, that regulates the carbohydrate metabolism. We've got aldosterone, which controls iron reabsorption in the kidneys and blood pressure. And lastly, the androgens regulate sexual characteristics and cell growth.
Next, we have a look at the pancreas. So this is a gland located behind the stomach. It releases hormones to control blood glucose levels and enzymes for digestion, functioning as an exocrine gland. Most of the pancreas is made up of the exocrine tissue secreting amylase, proteases, and lipases. There are small regions of endocrine glands amongst the exocrine tissue, and these are called the islets of Langerhans. The islets of Langerhans are made up of alpha cells and beta cells, and the alpha cells secrete glucagon, and the beta cells secrete insulin.
Quick 2025 updates to make it really mark scheme specific. You do need to know the name of the acinar cells, which are the examples of the exocrine tissues, and they secrete the amylases, proteases, and lipases.
And this takes us into the control of blood glucose concentration. The amount of glucose in your blood will increase after ingesting food containing carbohydrates or drinks containing carbohydrates, and it decreases following exercise or if you haven't eaten anything containing carbohydrates in a period of time.
Important update here for being mark scheme specific. throughout all of these slides coming up on blood glucose concentration, you do need to use the phrase blood glucose concentration, not blood glucose levels, to get the mark. So bear that in mind. If I say at any point in those slides blood glucose levels, make sure that you have it written down as blood glucose concentration.
To control the blood glucose concentration, the pancreas is one of the key organs. It detects changes in the blood glucose levels. The islets of Langerhans will then release insulin and glucagon to bring it back to its normal level. Insulin is released when glucose levels are too high in the blood, and it causes a decrease in blood glucose levels. Glucagon, on the other hand, is released when blood glucose levels are too low, and it causes an increase in blood glucose levels. Adrenaline is released by the adrenal glands when your body anticipates danger, and this results in more glucose being released from the hydrolysis of glycogen in the liver. So if you have a look at this negative feedback response, your blood glucose levels would increase after eating a carbohydrate-rich meal. This would be detected by the beta cells in the islets of Langerhans in the pancreas. The beta cells will then release insulin. We're going to go through how insulin then causes changes, but essentially the shorthand is liver cells will then become more permeable to glucose, and enzymes are activated to convert glucose to glycogen. That will then remove glucose from the blood and store it as glycogen in the liver cells. And as a result, your blood glucose levels decrease and go back within the normal limit. Now, alternatively, maybe you've just done lots of exercise and the glucose has been used up for respiration, so your blood glucose levels have now decreased. This is detected by the alpha cells in the islets of Langerhans in your pancreas. Alpha cells will then release glucagon, and the adrenal glands will release adrenaline. We'll have a look at what the second messenger model is shortly, but that will occur and activate enzymes to hydrolyze the glycogen within the liver cells back into glucose. That will then be released into the blood and increase your blood glucose levels back within the normal limit.
So if you do want to draw out or write down this flow diagram to help you to remember it, here's one that you can screenshot where you've got the actual key terms added in as well to show you which stages are glycogenesis, gluconeogenesis, and glycogenolysis as well.
So if we have a look at the control of insulin secretion, when there is a normal blood glucose concentration, the potassium ion channels within the cell membrane of the beta cells remain open, maintaining that minus 17 millivolt resting potential. If the blood glucose concentration increases, glucose enters the cell via glucose transporter. That absorbed glucose is used in respiration to make ATP, which binds to the potassium ion channels. This causes them to close and no more potassium ions can diffuse out of the cell, resulting in depolarization. The depolarization causes voltage-gated calcium ion channels to open, so calcium ions enter. And when the calcium ions enter, they cause secretory vesicles to move towards the cell membrane and release the insulin they contain by exocytosis. So the action of this released insulin then is the beta cells that are releasing it, and the insulin will cause a decrease in blood glucose levels in the three following ways. It will attach to receptors on the surface of target cells, which would be your liver cells. This changes the tertiary structure of the channel proteins embedded within the membrane, resulting in more glucose being absorbed by facilitated diffusion. More of those protein channels are also incorporated into the cell membrane so that more glucose is absorbed from the blood into the cells. And lastly, the binding of the insulin to the receptor activates enzymes involved in the conversion of glucose to glycogen. And this results in glycogenesis in the liver. So this here is just showing you part of that idea. We've got the insulin bound to the receptor on the cell surface membrane. The binding causes the vesicles which contain the glucose channel proteins to move towards the cell surface membrane. They will then fuse with the membrane and you then get more of these channel proteins embedding.
Let's take a look then at the action of glucagon. It is the alpha cells in the islets of Langerhans that detect when the blood glucose is too low, and in response it will secrete the hormone glucagon into the blood. And glucagon can then cause an increase in blood glucose in the following ways. Firstly, it can bind to the complementary shaped receptors on the surface of its target cells, which are the liver cells. When it binds to that receptor, it causes a protein to be activated within that cell. And that is going to be activating the protein into adenylate cyclase, which is then able to convert ATP into a molecule of cyclic or cyclic AMP, also known as CAMP. CAMP then activates another enzyme, which is a protein kinase, that can hydrolyze the glycogen into glucose, which is our glycogenolysis. It can also activate other enzymes which are involved in the conversion of glycerol and amino acids into glucose. So let's have a look at this second messenger model, which is one of the responses that we see from glucagon. Glucagon will bind to those complementary shaped receptors on the cell surface membrane of the liver cells, which are the target cells. Once bound, it causes a change in shape to the enzyme adenylate cyclase, which activates that enzyme. And as we said, that is the enzyme that can then catalyze the conversion of ATP into CAMP. And that is the second messenger. The first messenger is glucagon, because that causes the activation of this enzyme. Second messenger is cyclic AMP, because that causes the activation of this enzyme, which is the enzyme that is responsible for hydrolyzing glycogen into glucose. The role of adrenaline is linked to these fight or flight responses, basically meaning when you might be scared or anticipating dangers, you might have to suddenly run or fight to protect yourself. The adrenal glands are going to secrete adrenaline. And adrenaline will increase the blood glucose so that you have more glucose in the blood for respiration, so there's more ATP being produced for muscle contraction in case you do have to run or fight to protect yourself. So what happens here is, first of all, the second messenger model again. So that is when this time it's adrenaline binding to the complementary shaped receptors on the cell surface membrane of the target cells, which would be liver cells. It again activates the adenylate cyclase, which can convert ATP into cyclic AMP. The cyclic AMP then activates a protein kinase enzyme, and that can then hydrolyze glycogen into glucose. So basically, the cyclic AMP is going to be the second messenger because it's then activating an enzyme that can cause the hydrolysis of glycogen into glucose. So that is the second messenger model, exactly like we just saw for glucagon. It's triggered this time by the binding of adrenaline to adrenaline receptors on the cell surface membrane of the liver cells.
So in summary then, the roles of the liver, we have glycogenesis, and that's when we convert glucose into glycogen. And this occurs in the liver and it's catalyzed by the enzymes there. Glycogenolysis is the hydrolysis of glycogen to glucose, and this occurs in the liver due to the second messenger model. Gluconeogenesis is the creation of glucose from other molecules such as the amino acids and the glycerol. Diabetes is the inability to control your blood glucose levels. And type 1 diabetes is when you're unable to produce insulin or you don't produce sufficient amounts. And this typically starts in childhood as a result of an autoimmune disease where the beta cells have been attacked and destroyed, and therefore they're unable to produce sufficient amounts or any insulin. The treatment would therefore be injections of insulin. Type 2 diabetes is when the receptors on the target cells lose their responsiveness to insulin. And this usually develops in adults because of obesity and poor diet. It's controlled by regulating the intake of carbohydrates in your diet, increasing exercise, and sometimes insulin injections as well. So this insulin that we're talking about being used in the treatment of diabetes is produced by genetically modified bacteria. But in the future, diabetes could potentially be treated by using stem cells. And this is focusing on type 1 diabetes, where the autoimmune disease has damaged the beta cells. So you could use stem cells to replace those faulty B cells which can't produce enough insulin. The stem cells would probably have to come from embryos, which comes with ethical concerns around the destruction of the embryos. However, if it was successful, patients wouldn't have to constantly inject insulin or wait for a potential pancreas donor, and the stem cells, if they were from a cloned embryo of themselves, would be unlikely to be rejected. Next, we move on to plant and animal responses. Now, plants can't run away from animals trying to eat them or move to the shade if they're too hot like animals can. Therefore, plants have evolved to have a range of different responses to herbivore versus abiotic stress. Plants can defend themselves against herbivores using physical and chemical defenses. Physical defenses could include thorns, stings, spikes, barbs, and fibrous inedible leaves. Chemical defenses include tannins, alkaloids, and terpenoids. Alkaloids are nitrogenous, bitter-tasting chemicals, and alkaloids affect the metabolism of herbivores, resulting in death. Examples include nicotine, caffeine, cocaine, and morphine. Pheromones are also one of the chemical defenses, and these are chemicals that are released and affect the behaviour of other members of that species. Animal social behaviour are affected by pheromones, but plants use them to communicate about danger. Volatile organic compounds act like pheromones for plants. So some examples include trees can release these pheromones when they are being attacked by an insect. And the release of that pheromone can cause neighbouring trees to produce callose to help protect them against the insect attack. Another type of response is the folding in response to touch. And the Mimosa pudica is a rare example of a plant that can move to scare off predators. The leaves of this plant fold when they are touched, and the movement can scare animals and brush insects off them.
Interrupting here to make you aware of a specification change that did happen for the 2025 spec. You now only need to know pheromones and the alkaloids for the chemical defences. You don't need to know any of the others that I had on the slide shown.
In contrast, if we have a look at the responses to abiotic stresses the plants are exposed to could include high winds, excess water, a lack of water, temperature changes, change in day length, and changes in salinity, which is the salt concentration. The responses that the plants have include leaf loss. So trees will lose their leaves in countries that have cold winters. When it gets cold and the daylight hours decrease, the rate of photosynthesis decreases. And at this point, it's more energy efficient for plants to lose their leaves. Day length sensitivity also occurs. Photoparedism is the term for plants being sensitive to a lack of light. Plants are sensitive to how long it's dark for, and when they detect the dark periods are shorter, when there is longer daylight hours, it will cause the leaves to bud and flowers will bloom after winter. Next, we look at abscission, and this is when light levels decrease in autumn and winter. Ethene switches off genes for enzymes that digest and weaken the cell at the abscission zone. So there's a separation layer in a leaf petiole. This can cause the leaf to separate from the plant, leaving a waterproof scar behind to protect the rest of the plant. Some plants also contain chemicals which act as a natural antifreeze, and this is what prevents the cytoplasm from freezing when it's really, really cold. There is also stomatal control, and this is when the stomata can open and close in response to different stimuli. The evaporation of water out of the stomata provides a cooling effect to the plant, and that opening and closing of the stomata can be controlled by the hormone ABA in response to temperature stress. Tropisms is another example of responses in plants, and in this way the plants respond via growth in response to a stimulus. They can be positive or negative. Positive is when the plant would be growing towards the stimulus. Negative is when it grows away from the stimulus. And they tend to respond to light, gravity, and water stimuli. Tropisms are controlled by specific growth factors, and one key example is IAA. IAA is a type of auxin that can control cell elongation shoots and inhibit the growth of cells in the roots. It's made in the tip of the roots and the tip of the shoots, but it can diffuse to the other cells.
An edit I want to make you aware of here is, for the OCR exam board, they actually prefer you to say auxin rather than IAA. So I've explained in those slides, but you can screenshot this slide to make your notes from, and then also this one where it's been corrected to say auxin. So using the word auxin instead of IAA for OCR.
So if we have a look at phototropisms in the shoots first of all. Light is needed for the light-dependent reactions in photosynthesis. So it's important that the plant is able to have as much exposure to sunlight as possible. So the plant will grow and bend towards the light, and this is positive phototropism. The way that this happens is, in the shoot tip, IAA is produced, and in shoots, exposure to IAA causes cells to elongate and therefore plant growth. The IAA can diffuse to those other cells and cause the growth in the other cells as well. But if you have unilateral light, meaning from one direction rather than equally distributed, the IAA will diffuse to the shaded side. And that's what we can see here. When the sun is now, or the light source is on this side, it causes the IAA to diffuse to the shadier side. And that means the cells on that side will elongate more, grow more, and that's how we get this growing and bending towards the light source. The opposite is happening in roots. Roots do not photosynthesize, so they do not require light energy. But it is beneficial if they can grow down towards water sources and anchor deep in the soil. In roots, a high concentration of IAA inhibits cell elongation, causing root cells to elongate more on the lighter side, and so the root ends up bending away from the light. And this is negative phototropism. In gravitropism in the shoots, the IAA will diffuse from the upper side to the lower side of a shoot. If a plant is vertical, this causes the plant cells to elongate and the plant grows upwards. But if you were to put a plant on its side, like in this example, that means the IAA will diffuse to the lower side with gravity, and that means the cells on the lower side will elongate and your shoot bends and grows upwards. So this is negative gravitropism. In the roots, the IAA diffuses to the lower side of the root with gravity, but as we said, in the roots, IAA prevents or reduces that elongation. So that means the cells on the top where there wasn't IAA will elongate more, and that's why the root bends down, and that is your positive gravitropism. Plants also produce hormones, and the hormones control a range of responses in the plants, such as the ripening of fruit, germination of seeds, lengthening of stems, and when the leaves drop. And here are 4 examples of hormones in plants that you need to know about and their roles. So these would make really good flashcards. I recommend pause and turn those 4 hormones and what they do into flashcards.
We're now going to look at apical dominance. And auxins stimulate the growth of shoots, particularly the main shoot, which is known as the apical. Auxin results in that apical dominant, which means one main shoot growing, which inhibits the growth of the lateral shoots due to the high concentration of auxin at the apical shoot point. The higher the concentration of auxin, the stronger the apical dominance, and therefore the less growth there'll be of those lateral shoots, meaning the ones coming out of the sides. So if we look at the evidence for this, if the apical shoot is removed, which we said is the one that contains the auxin producing cells in the tip, then the lateral shoots start to grow faster. When auxin is then artificially applied, the lateral shoot's growth rate decreases again. This shows the presence of auxin suppresses the growth of lateral shoots. Next, we're going to have a look at gibberellins in relation to seed germination. A seed starts to germinate when it absorbs water, and this activates the production of the hormone gibberellins, or the plant growth factor. The gibberellins cause enzymes to be released that can break down the food stores in the seed so that the embryo plant can use the food to respire and therefore make ATP. Evidence suggests that gibberellins cause this to happen by switching on genes that code for amylases and proteases, so those 2 enzymes. Evidence also indicates that abscisic has an antagonistic effect, and that it is the levels of these 2 hormones that control when a seed germinates. So here's the evidence. There are experiments that have been conducted using mutant plant varieties which do not have the gene that codes for the gibberellins. The mutant plant seeds did not germinate, but when they were then exposed to an external source of gibberellins, the seeds did germinate. The other experiment then is using gibberellin biosynthesis inhibitors, which also showed that plants were unable to make gibberellins, or when they were unable to make the gibberellins, their seeds did not germinate. And when these plants were then given gibberellins, the seeds did then germinate. Gibberellins also have a role in stem elongation. So they are a collection of hormones that help plants grow by stimulating elongation in the stem. The higher the concentration of gibberellins, the more elongated the stem will become. So the evidence for this is that dwarf varieties of plants have very low levels of gibberellins, and this is often due to a mutation in a gene involved in the synthesis pathway of the gibberellins. Scientists have also experimented by treating these dwarf varieties with an external source of gibberellins, and that did then result in them growing to the same height as the non-dwarf varieties. There's also horticulturists and farmers that apply gibberellins to shorter plants to stimulate plant growth. So this knowledge then of the plant hormones has been used commercially. For example, ethene is used to control the ripening of fruits. Unripe fruits can be picked and transported whilst they're still firm, and then sprayed with ethene before it's sold so that they then do ripen up. You also have rooting powders which are used to encourage the growth of new roots from plant cuttings, and auxins are also used in those rooting powders for this. And lastly then, the auxins have also been used as weed killers. You can spray auxin over weeds, which causes them to grow so quickly that their stems give way and the weeds end up breaking and dying.
Next, we move on to animal responses, and that takes us to the mammalian nervous system, which is made up of the peripheral nervous system and the central nervous system. The peripheral nervous system includes the receptors, sensory and motor neurons, and the central nervous system is the coordination centre, which includes the brain and the spinal cord. The nervous system can be categorised into the autonomic or the somatic nervous system. The autonomic nervous system works constantly and subconsciously. This includes activities such as digestion, which you have no conscious control over. The somatic nervous system is consciously controlled. This is voluntary and is when you decide to move, for example, when you choose to stand up. The human brain is made up of billions of neurons and it coordinates responses. The key structures that you need to know are the cerebrum, the cerebellum, the medulla oblongata, and the hypothalamus and the pituitary gland, which we came across those two earlier in this video. The cerebrum is the largest part of the brain, and it's the outer layer, sometimes known as the cerebral cortex. It's made up of many folds and it splits into two hemispheres. The functions range from controlling conscious thoughts, language, intelligence, personality, and high-level functions and memory. The cerebellum, which is the part towards the back that looks like a mini cauliflower, is responsible for coordinating movement, muscles, and balance. Medulla oblongata is just above the spinal cord, and it's the centre of control of unconscious activities, such as breathing and heart rate. The hypothalamus is the small part of the brain responsible for homeostasis, such as temperature and water balance. The pituitary gland is a small lobe structure known as the master gland because it secretes many hormones to coordinate several responses, such as the estrous cycle and osmoregulation.
Just interrupting on this slide to let you know that I've added in some extra details here from the mark schemes that came out this year. So here you have some extra details, so screenshot and use this slide for the extra details that they're after now for the 2025 spec for that level of detail.
A reflex is a type of animal response, and it's a rapid, automatic response to protect you from danger. In a reflex arc, there are only 3 neurons. You'd have a sensory neuron, a relay neuron, and a motor neuron. And because you've only got 3, that means you'd only have 2 synapses. So what makes it rapid is the fact there are only 2 synapses, and the synapses slow down the speed because it involves the diffusion of the neurotransmitters. As there's no conscious decision involved, the response is also rapid and it prevents the brain being overloaded with situations to decide responses. Once the stimulus is detected by the receptor, an impulse is passed along the sensory neuron to a relay neuron. The relay neuron passes the impulse onto a motor neuron, which is connected to an effector. For example, if the stimulus is a hot object, the effector would be a muscle in your hand or your arm, and the response would be for the muscle to contract to move you away from that danger. Other examples include the knee-jerk reaction and blinking. Another response is the fight or flight response in animals. When animals are exposed to a potential threat to their survival, a series of automatic responses are triggered to prepare the organism to either fight to survive or run away from the danger. The autonomic nervous system detects the potential threat, sending an impulse to the hypothalamus. This results in more impulses being transmitted along the sympathetic nervous system and the adrenal cortical system. The effectors are the adrenal glands, which will release more adrenaline and noradrenaline. These release of hormones trigger the hypothalamus to stimulate the release of adrenocorticotropic hormones, or ACTH, from the pituitary gland. Now, the action of adrenaline we've actually come across already when we looked at the role of this in the blood glucose concentration, but we've got the roles summarized here again.
Just an edit to this video to make it more mark scheme specific. You need to name adrenaline as the first messenger, and you then need to know that it is this enzyme that converts ATP into cyclic or cyclic AMP. So next then we look at the control of heart rate, and both the endocrine and the nervous system can affect the heart rate. Whilst the heart has an inbuilt pacemaker, the sino-atrial node or the SA end, and it is myogenic, meaning it can beat without the input of the nervous system, it can also be affected by both the endocrine and the nervous system in order for the heart rate to respond to stimuli. So in other words, it can beat without the input of the endocrine and nervous system, but the speed or the heart rate is determined by these two systems. Hormonal control is also involved because it's the endocrine system. And the hormone adrenaline has three effects on the heart. It increases the heart rate, it increases the stroke volume, and it increases the cardiac output. So if we look at the nervous system control of the heart rate, the cardiovascular center in the medulla oblongata in the brain is the part that controls heart rate. And this is via the autonomic nervous system, meaning you don't have any conscious control. The heart and the medulla oblongata are connected via two nerves that are connected to the SAN, which is that pacemaker, the sino-atrial node. Increases in heart rate are caused by impulses sent via the accelerator nerve in the sympathetic nervous system, and decreases in heart rate are caused by impulses sent via the vagus nerve in the parasympathetic nervous system.
The heart rate changes in response to pH and blood pressure, and these stimuli are detected by chemoreceptors if it's a change in pH, and pressure or baroreceptors if it's a change in blood pressure. And these receptors are found in the aorta and the artery directly after that, which is the carotid artery. So if we have a look at the response to pH, the pH of the blood will decrease when there is a lot of respiration because carbon dioxide is produced, or lactic acid, and both of those will decrease the pH of the blood. Now that carbon dioxide and lactic acid have to be removed rapidly so they don't denature enzymes. And this is achieved by the response here, an increase in the heart rate, and that is because there'll be more impulses via the sympathetic nervous system to the SAN in the heart, which causes the heart rate to increase and therefore pump the blood more rapidly to the lungs, where carbon dioxide can diffuse out at the alveoli. In response to pressure, if the blood pressure is too high, this can cause damage to the walls of the arteries, and it's important to put mechanisms in place to reduce the blood pressure. This results in more impulses via the parasympathetic nervous system to decrease the heart rate. If the blood pressure is too low, there may be insufficient blood supply of that oxygenated blood to respiring cells or insufficient removal of waste. So this results in more impulses via the sympathetic nervous system to increase the heart rate. Next, we move on to looking at muscles.
Now there are three types of muscles that you need to know about. So we'll go through these three on this slide. Skeletal muscles, and most muscle is skeletal, and this is the muscle that's attached to the skeleton and therefore responsible for causing movement of the skeleton. It's made out of cylindrical shaped cells which join to form multinucleated myofibrils, which means it contains lots of nuclei. They have a striated pattern when stained and viewed under the microscope. There's also cardiac muscle, and the heart contains cardiac muscle and it's used to pump blood. It's myogenic, which means it doesn't require an input from the nervous system to cause it to contract and relax. And the cells are branched to allow contraction across the whole of the atrium or ventricles, and the cells are uninucleated, meaning it contains only one nucleus. They have a striated pattern when stained under the microscope as well. Lastly, we have the involuntary, or also known as smooth muscles. This is the muscle that lines organs and blood vessels, and by contracting and relaxing, it causes the movement of the contents of an organ or the blood vessel. Examples include controlling the diameter of the arteries, arterioles, bronchi and bronchioles, as well as controlling the size of the pupils and peristalsis in the digestive system. These cells are also uninucleated, meaning there's only one nucleus, spindle-shaped, unstriated, stained and viewed under a microscope.
A neuromuscular junction is where we have a neuron meeting the muscles, which would be acting as your effector in the response arc. You might need to know differences between the neuromuscular junction and the cholinergic synapse. There are many similarities because you still have this same idea of the neurotransmitters being released, but some of the key differences are the neurotransmitter is binding to receptors on the muscle fibers rather than on the postsynaptic neuron. The other differences are written out here, and this would make quite a good flashcard to summarise these differences between your neuromuscular junction and your cholinergic synapse.
This is a 2025 update where I have actually added quite a bit more detail to this text box for you to be aware of. So the neuromuscular junction is the junction between a motor neuron and a muscle fibre, similar to a synapse. But you also need to know all of this detail here. So when an impulse arrives at the end of a motor neuron, a neurotransmitter passes across that neuromuscular junction and it binds to receptors on the sarcolemma, which is the membrane of the muscle cell, which we can see here. This causes the receptors to open, sodium ions will then move in, and the membrane becomes polarized. The wave of depolarization is passed down T tubules, causing the sarcoplasmic reticulum to release calcium ions, which leads to the muscular contraction.
Focusing on the skeletal muscles then, muscles act in antagonistic pairs against an incompressible skeleton, and that's what creates movement. This could be automatic as part of a reflex response, or it could be controlled by conscious thought. Myofibrils are made up of fused cells that share a nuclei and cytoplasm, which is known as the sarcoplasm, and there are a high number of mitochondria. These myofibrils are made up of sarcomeres, and we can see here one section, the sarcomere in a myofibril. And a sarcomere is made up of the proteins actin and myosin. The muscle fibers are made up of millions of myofibrils, and those collectively bring about the force to cause movement. Those myofibrils are made up of two key proteins, the myosin and actin, and that is what forms the sarcomere. So this here is showing you the actin and the myosin within your sarcomere. And they are layered on top of each other in these different bands. And you could be asked to talk about what happens to the width of the bands when a muscle contracts. Your A band is the length of the myosin. Your H zone is where you have just myosin with no actin overlapping it. The I band is where you have just actin with no myosin overlapping it. The Z line is the barrier or the end of one of the sarcomeres. So when your muscles contract, we'll look at the sliding filament theory in the next slide, but essentially you are sliding these fibers closer together. So that means that your A band will always remain constant because the myosin isn't getting any thicker or thinner. It's simply sliding the actin closer together. So the A band remains constant, but the H zone decreases inside because the actin is now moving closer together. That also means that the I band has decreased and our Z lines are now closer together. And this here is showing you that under a microscope, and you can see that those Z lines have become closer together. So what is actually causing this to happen is explained by the sliding filament theory. When an action potential reaches the muscle, which is the effector, it stimulates a response. Calcium ions enter and cause the protein tropomyosin to move and uncover the binding sites on actin.
Quick 2025 edit here, just adding in a bit more detail on this second bullet point. Now that the calcium ions enter and bind to the protein troponin, causing a change in the shape of the protein tropomyosin, which is the protein that blocks the binding sites on actin for the myosin head during relaxation. So that causes it to move and uncover the binding sites.
So we can see here this tropomyosin shown in yellow, and it is blocking some of those binding sites. But when calcium ions enter, it moves the tropomyosin out of the way, so all of these binding sites are now revealed. On the myosin, we've got these structures called the myosin heads, and ADP and PI are attached to it. And whilst ADP is attached to the myosin heads bind to the actin to form this cross-bridge structure. That binding creates this angle and tension, and as a result, the actin filament is pulled and slides along the myosin. And in doing so, the ADP molecule is released. An ATP molecule then binds to the myosin head, and that causes it to change shape and detach from the actin. Within the sarcoplasm, there is an enzyme ATPase, which is activated by the calcium ions to hydrolyze that ATP back into ADP and PI. And that releases enough energy for the myosin head to return to its original position. This entire process continually repeats, sliding the actin closer and closer together, which is your muscle contracting. And that will continue to happen as long as calcium ions are being released by the sarcoplasm, which means that the tropomyosin is being moved out the way. The role of ATP and phosphocreatine in this are important also. Active muscles need a high concentration of ATP, and that is because of their role in the sliding filament theory. The chemical phosphocreatine, which is stored in muscles, assists this by providing the phosphate to regenerate ATP from ADP. We next move on to photosynthesis, and we start by looking at the structure of a chloroplast. So your chloroplast is a double membrane-bound organelle, and within that we then have these thylakoid membranes that are highly folded and form these stacks called a granum, or grana for plural. And those thylakoid membranes have lots of photosynthetic proteins such as chlorophyll embedded within them, and also electron carrier proteins which are used in the light-dependent reactions. There's also the stroma, which is the fluid centre which contains the enzymes involved in the light-independent reactions. The inner and outer membranes, just like other plasma membranes, control what can enter and leave the organelle. So looking at chlorophyll in a bit more detail, chlorophyll is located in the photosystems on the thylakoid membrane, and those are the proteins embedded within the membrane. And chlorophyll is a mix of different proteins that can absorb a range of wavelengths of light. There are 5 key closely related types of pigments, but chlorophyll A is the most abundant. Below are the other types that you need to know about. So chlorophyll A, which is bluey green and in all plants, but there's also chlorophyll B, carotenoids, xanthophylls, and phaeophytins. The reason that it's an advantage to have a range of pigments is they each absorb a slightly different wavelength of visible light, and this maximizes the spectrum of visible light that gets absorbed, and therefore overall it increases the amount of light energy absorbed. And that is what this graph over here demonstrates. Here we've just got chlorophyll A and B represented, showing you the wavelengths of light that they absorb, and which ones must be reflected because there is a low absorption rate. So that means all of this visible light is being reflected and wasted. By having multiple additional pigments, that would increase the wavelengths of light that are absorbed.
This is an edit here that I'm adding in for OCR to be mark scheme specific. You only need to know these 4 types of pigments, not 5. So these are the 4 that are needed for the OCR exam board. So chlorophyll B, xanthophylls and carotenoids are known as the accessory pigments and are all embedded in the thylakoid membrane to form a light harvesting system. The light harvesting system is where light energy of different wavelengths is absorbed, and this energy is then transferred to the reaction centre. The reaction centre is where chlorophyll A is stored, which is the primary pigment, and it is where the light-dependent reactions occur in photosynthesis. The light harvesting system and the reaction centre make up a photosystem. Now, all of these photosynthetic pigments that we've discussed can be isolated using chromatography, for example, thin layer chromatography or TLC. The pigments are added to a TLC plate, which is placed in a solvent. The solvent dissolves the pigments, and the more soluble the pigment is, the further up the plate it will move. This can be converted into an RF value, and that can then be used to compare with known RF values for particular pigments to be able to identify which pigments you have in your sample. And you'd use this formula here to work out the RF value.
So now let's have a look at the actual reactions, starting with the light dependent stage. This is the first stage of photosynthesis and involves harvesting energy from light. The purpose of this stage is to harvest that light energy, use it to split water, and then it's resulting in the creation of ATP and reduced NADP. And those two molecules are needed in the light independent reactions. So the light dependent stage happens on the thylakoid membrane, and it involves 4 key steps: non-cyclic photophosphorylation, cyclic photophosphorylation, photolysis or photolysis, and chemiosmosis. So non-cyclic photophosphorylation, this involves the 2 photosystems, photosystem 1 and photosystem 2. Photosystem 2 is the first of the 2 photosystems to be used. Here you'll have pigments which will absorb the light with a wavelength of 700 nanometers. Photosystem 1 is then used, and it has pigments that can absorb light with a wavelength of 680 nanometers. So that energy, that light energy that is absorbed, causes electrons from within the reaction centers to be excited and released. So this light energy that is absorbed in that photosystem 2, it will then cause electrons from the chlorophyll to be excited and released. They'll be released from photosystem 2. You'll also have some being released from photosystem 1, and those electrons then move along the proteins embedded within the thylakoid membrane, and that is your electron transport chain. As those electrons move along, it results in energy being released, and that is going to link to what we're talking about in chemiosmosis in a couple of slides' time. But ultimately, it results in the production of ATP. The electrons lost from photosystem 2 are replaced by electrons from photolysis, which is coming up in the next couple of slides. And the electrons lost from photosystem 1 are replaced by the electrons that are moving along the transport chain from photosystem 2. At the very end of the electron transport chain, which we can see here, those electrons are picked up along with protons by NADP to form reduced NADP or NADPH. That reduced NADP and ATP made in this process are then needed in the light independent reactions. Cyclic photophosphorylation, in contrast, some of those electrons that are released from photosystem 1 are not picked up by NADP and instead are recycled back to photosystem 1. The transport of electrons still results in ATP production though, because as those electrons are moving, they're still going to be releasing energy. And that is through chemiosmosis, which we'll be coming to very shortly. So in this way, cyclic photophosphorylation does result in the production of ATP, but not the production of reduced NADP. Now we mentioned photolysis of water, replacing the electrons lost from photosystem 2. So photolysis or photolysis literally means light splitting. So we've got light splitting water. And that's what happens. Light energy is absorbed, and that energy will then split water into oxygen, electrons, and protons. Those protons are picked up by the NADP to form NADPH. The electrons we just talked about are picked up by photosystem 2 to replace the ones that were lost in the electron carrier chain. And the oxygen isn't used, so that can be used in respiration, or it'll just diffuse out of the stomata. Finally then, in the light-dependent reactions, we have chemiosmosis. And here we can see the involvement of photosystem 2 and 1 again. So the electrons that are excited and released by the chlorophyll within photosystem 2, we said move along the embedded proteins in that electron transport chain. As they move from protein to protein, they release some energy, and that energy is used to actively transport protons from the stroma into the space within the thylakoid, known as the thylakoid lumen. As a result, we end up with lots of protons within the thylakoid lumen compared to the stroma, and this creates an electrochemical gradient. Those protons then move down their electrochemical gradient through the only protein that they're complementary in shape to bind to, which is ATP synthase. And as they move through, that enables that enzyme to catalyze the phosphorylation of ADP and PI into ATP. So that is how we create ATP through our non-cyclic and cyclic phosphorylation in chemiosmosis. And then finally, that reduced NADP, at the end of that electron transport chain, the electrons and the protons that have returned are picked up by NADP to make our reduced NADP or NADPH. So next we move on to the light-independent reactions, and this is also known as the Calvin cycle. Now this occurs in the stroma, which is the fluid centre of the chloroplast, which contains the enzyme Rubisco, which catalyses one of the key steps in this reaction. And because there's an enzyme, it's temperature sensitive. The Calvin cycle uses carbon dioxide, and it's going to use the reduced NADP and ATP from the light-dependent stages to form a hexose sugar. The ATP is going to be hydrolyzed to provide energy for these reactions, and the reduced NADP donates the hydrogen to reduce GP into TP. So let's have a look at this in more detail. We start off by seeing how the Rubisco is involved, and carbon dioxide enters the cycle by reacting with a 5-carbon compound represented by these 5 yellow circles, and carbon dioxide and RUBP react together to form 2 molecules of GP, catalysed by Rubisco. So we now have 6 carbons in total, but it's split into 2 3-carbon compounds. That GP is then reduced into 2 molecules of TP, but the reduction is where we see the use of reduced NADP. So hydrogen is released from the NADP, picked up by GP to reduce it. And this stage requires energy, and that's why ATP is needed at this point. Some of the carbon, in fact it's 1 carbon, is lost from those 2 TP molecules every cycle. And that goes towards making a hexose sugar, or useful organic substances. So that means you would need 6 turns of the Calvin cycle to make a hexose sugar. And each time when 1 carbon is removed, that means you have 5 carbons left, but we need to join them back together and regenerate the RUBP so the cycle can continue. And energy is required to regenerate that RUBP. So although glucose is the product from those hexose sugars, that is a monosaccharide, and it can join to form disaccharides, such as sucrose through condensation reactions, or polysaccharides such as cellulose for the cell wall, or even stored away as starch. It can also be converted into glycerol, and therefore combine with fatty acids to make lipids, or it can be used to convert into amino acids to make proteins. Now within photosynthesis there are limiting factors, and a limiting factor is anything that reduces the rate of photosynthesis. And that could be temperature, light intensity, or carbon dioxide concentration. Temperature, because it's an enzyme-controlled reaction. Light intensity, because light is required for the light-dependent stages. And carbon dioxide, because that is needed for the Calvin cycle, or the light-independent reactions. Now, light intensity in carbon dioxide, at high enough levels, the rate plateaus, because at that point, there'd be something else limiting the rate of reaction. Whereas temperature, because it's due to enzymes, you'll get this increase in rate, but then if it gets too hot, the enzymes denature and the rate would drop back down to 0 or to a much lower rate.
Quick 2025 edit, and that's just here the addition of water stress, as well as a limiting factor, which means a lack of water. So here are the effects of those limiting factors. Carbon dioxide is a reactant in the light independent stage, and therefore the concentration affects the rate. If there's a low concentration of carbon dioxide, that means there'll be less carbon fixation within that Calvin cycle. So the levels of RUBP will build up. Less carbon fixation will then prevent GP and TP being made, so their levels will decrease. Next up, we will look at water stress. This will cause abscisic acid to move to the guard cells, closing the stomata to prevent water loss by transpiration. This will then prevent carbon dioxide entering the stomata by gas exchange, and there'll be less carbon fixation. So again, we'll get levels of RUBP will build up, and we'll have lower levels of GP and TP. Temperature affects the enzyme-controlled reactions in photosynthesis, such as fixation of carbon dioxide by the enzyme ribisco. Less carbon fixation means the levels of RUBP will build up, and again, less carbon fixation prevents GP and TP from being made, so those levels will decrease. Lastly, the light intensity affects the rate because it's needed for the production of ATP and reduced NADP in the light dependent stage. If there's less ATP and less NADPH or reduced NADP, that will prevent GP from being converted to TP. So that will mean GP levels will increase and TP and RUBP levels will decrease because you don't have the GP being converted to TP and you don't have TP being regenerated into RUBP.
For maximum photosynthesis and therefore plant growth, common agricultural practices incorporate techniques to remove limiting factors. And that could range from using artificial lighting to maximise the light intensity, or heating, or even burning fuels to get more carbon dioxide. But the extent to which each technique is used needs to be considered in terms of profit. Because if the extra growth from paying for all of these conditions does not exceed the cost, then it's not cost effective. Finally, we move on to respiration.
So ATP is a molecule that you learned about in the biological molecules topic earlier on in the course, but you now need to know it in a context. So you might be asked to put in a context, so for example, state the uses of ATP made in respiration in bacteria cells or liver cells, for example. So you need to learn a list, just like the one we've got here, to be able to select which one of those uses would be appropriate for the particular question you might be given. So we've got movement. ATP can be used to provide the energy for movement, for example, muscle contraction or movement of a flagella in bacteria. Active transport, endocytosis and exocytosis, cell division, synthesis of biological molecules, because in proteins, for example, you need ATP to form the peptide bonds between amino acids, DNA replication, and also maintenance of body temperature.
And in aerobic respiration, there are 4 key steps. Glycolysis, which happens in the cytoplasm of the cell, link reaction, which happens in the mitochondrial matrix, the Krebs cycle, which happens in the mitochondrial matrix, and oxidative phosphorylation, which happens on the inner membrane of the mitochondria, which is also known as the cristae. So the mitochondria is also a double membrane bound organelle, but the inner membrane is what folds in this organelle to create that large surface area with lots of proteins embedded in it for this final stage, oxidative phosphorylation. We're going to start though with glycolysis, and this happens in both aerobic and anaerobic respiration in the cytoplasm. And glycolysis involves 3 key steps: phosphorylating glucose to glucose phosphate using ATP, the production of triose phosphate, and oxidation of triose phosphate to produce pyruvate with a net gain of ATP and reduced NAD. So this here is just demonstrating those different things we just bullet pointed. We start with our glucose, a 6-carbon compound, and then we are phosphorylating to create glucose phosphate or hexose phosphate.
This is a really important update, so I'm going to put this at the start of this section, and I'm going to repeat this exact same clip at the end just to remind you that for the OCR spec, you need to call it hexose bisphosphate. So there should be a bis in front of the phosphate. So hexose bisphosphate is the name of the molecule that you need to know for the result of phosphorylating glucose.
So we've used 2 ATP molecules, hydrolyzed them both to release the phosphate group, and that then binds to the glucose, so it's phosphorylated, and that makes that glucose molecule gain energy and become more reactive. And for that reason, it then splits into 2 molecules of triose phosphate. That triose phosphate then undergoes oxidation, and we can tell it's oxidation because we have the coenzyme NAD picking up a hydrogen from the triose phosphate. And the fact that hydrogen has been lost from the triose phosphate means it's oxidized, and the NAD has picked up that hydrogen, so it is reduced. And that happens on both of the triose phosphate molecules. And that's why 2 molecules of NADH are made. And this stage also produces ATP. And each of these oxidation reactions will create 2 ATP, so that's 4 ATP in total. The final carbon compound that is made is pyruvate, a 3-carbon compound, and we have 2 of those. So in summary, from 1 glucose molecule, we've made 2 pyruvate molecules, we have 2 NADH, and we have a net gain of 2 ATP. So although 4 were made, 2 were used.
This is a really important update that for the OCR spec, you need to call it hexose bisphosphate. So there should be a bis in front of the phosphate. So hexose bisphosphate is the name of the molecule that you need to know for the result of phosphorylating glucose.
The pyruvate and the reduced NAD that were created in glycolysis are both then actively transported into the mitochondria and used in later stages. In the link reaction, that pyruvate is oxidized further into acetate, and NAD will pick up the hydrogen that is released when pyruvate is oxidized to make another molecule of reduced NAD or NADH. There is also decarboxylation, which means the removal of a carbon molecule, and that's how we get carbon dioxide. So acetate, which is formed, is a 2-carbon compound. That acetate combines with the coenzyme A and produces acetyl coenzyme A. And this has to happen so that the coenzyme is able to assist in allowing the acetate to enter the next stage, which is the Krebs cycle. So from 1 molecule of glucose, in the link reaction, we would get 2 acetyl-CoA, 2 carbon dioxides, and 2 reduced NAD. And that's because for 1 glucose molecule, we got 2 pyruvates being made, so the link reaction would happen twice. Then we get to the Krebs cycle, and we can see here how the link reaction is leading into the Krebs cycle. We've got the acetyl coenzyme A, and that is going to combine with 4-carbon molecule known as oxaloacetate. And when those two combine, we then get a 6-carbon compound, which is citrate. And once we form citrate, the coenzyme A is released, and that can be reused again in the link reaction. There is then a series of redox reactions that occur, which generates these reduced coenzymes and ATP through substrate level phosphorylation, and also this carbon dioxide that is lost. So we'd have 2 lots of carbon being released to make 2 carbon dioxide molecules. We have 1 ATP being formed. We have 3 reduced NAD being produced, and 1 reduced FAD coenzyme being produced. So that is our series of reactions that are happening in this stage. There are actually quite a lot of intermediates. So for example, you would have a 5-carbon compound, and then another carbon dioxide would be released, and then you get the 4-carbon compound, but you don't actually need to know all of the intermediates in this stage, rather all of the products that are coming out of the cycle. So in one cycle, we create 3 reduced NAD, 1 reduced FAD, 1 ATP, and 2 carbon dioxides. But per glucose molecule, because there'd be 2 pyruvates, there'd be 2 acetyl-CoA, and therefore the cycle happens twice. All of those are multiplied by 2. So in total from these first 3 stages, we have 10 reduced NAD and 2 reduced FAD. And these coenzymes are now all going to be used in the final step, which is oxidative phosphorylation. And this is the step where most ATP is made. Now this stage is chemiosmosis, so it's almost identical to what we just went through in photosynthesis.
It involves an electron transport chain, and I do just want to point out that I have edited this bit because it used to say transfer, but you have to say electron transport chain.
The movement of proteins across an inner mitochondrial membrane, and is catalyzed by the enzyme ATP synthase. So your reduced coenzymes that were all produced and are in the mitochondrial matrix, dehydrogenation happens, meaning the hydrogen is removed, and that hydrogen is then split into electrons and protons. The electrons are picked up by proteins embedded in that inner mitochondrial membrane, passed along the electron transfer chain. That releases the energy to actively transport the protons to create an electrochemical gradient. They then move down their electrochemical gradient through ATP synthase, and that enables ATP synthase to catalyze the phosphorylation of ADP into ATP. So that's how we make ATP. And because there were so many reduced coenzymes, we get lots of hydrogen ions and therefore lots of ATP. Lastly, at the end of that electron transport chain, those electrons have to be collected so that that chain can continue. And that is the role of oxygen. Oxygen is the final electron acceptor. It picks up the electrons and some of the protons that are within the matrix to form water. Next, then we look at anaerobic respiration, and this is respiration in the absence of oxygen, and it occurs in the cytoplasm of the cell only. The pyruvate is produced in glycolysis in exactly the same way that we just went through. But instead of it being oxidized in the mitochondrial matrix into acetate, it stays in the cytoplasm and it gets reduced to form ethanol and carbon dioxide in plants and microbes, or lactate in animals. And it does this by gaining the hydrogen from the reduced NAD that was created in glycolysis. That then reoxidizes NAD so it can be reused in glycolysis to make sure more ATP is continued to be produced. So this here is just demonstrating that. We've got glycolysis occurring, and then that pyruvate is reduced to form lactate using one of these reduced NAD. And that then removes the hydrogen because the pyruvate's picked it up, and that NAD can be recycled and reused. So although we're producing lactate, which is lactic acid when it's dissolved, which is toxic and causes harm, that still has to happen so that glycolysis can continue to happen to make at least some ATP.
Then if we have a look at anaerobic respiration in plants and microbes, this involves first of all the decarboxylation of pyruvate. So that is the pyruvate produced in glycolysis is going to have a carbon removed to form carbon dioxide to make ethanal. And then the ethanal is reduced to form ethanol by gaining the hydrogen from the reduced NAD. And this produces ethanol, oxidized NAD, and carbon dioxide. And that NAD can then be reused in glycolysis so that you can still get ATP being produced. So next then, if we have a look at respiratory substrates, glucose is the most common respiratory substrate broken down to release ATP in respiration. But it is not the only respiratory substrate. Respiratory substrates are organic molecules that can be used in respiration to produce ATP. So that means it can also include other carbohydrates, lipids, and proteins. So carbohydrates are used in glycolysis, for example glucose, or starch in plants and glycogen in animals, because starch and glycogen can both be hydrolyzed to release glucose. Lipids can be hydrolyzed into glycerol and fatty acids, and the glycerol can be converted into triose phosphate and can enter glycolysis to make pyruvate. Fatty acids can combine with the coenzyme A and be converted into acetyl coenzyme A to enter the Krebs cycle. Proteins can be hydrolyzed into amino acids, which are then deaminated to release keto acids, which can be used in the Krebs cycle. The respiratory quotient, or RQ, is the ratio of carbon dioxide molecules produced compared to the oxygen molecules taken in during respiration, and it can be calculated by doing carbon dioxide produced divided by oxygen consumed. There are some specific values of the RQ that indicate the type of substrate respired or even the type of respiration. So carbohydrates produce an RQ of 1, and that'd be if it's aerobically respired, because the carbon dioxide produced should equal the oxygen consumed, which is why you get an RQ of 1. Lipids produce an RQ of 0.7. Proteins produce an RQ of between 0.8 and 0.9. And if it's anaerobic respiration, the value will be greater than 1 because you don't have oxygen being consumed, but you do have carbon dioxide being produced. A respirometer is a device that can be used to measure the rate of respiration of living organisms by measuring its rate of exchange of oxygen and/or carbon dioxide. They allow investigation into how different factors could affect the rate of respiration. The organism the respiring organism that is, will be suspended in a gauze mesh in a sealed boiling tube above either a carbon dioxide absorber, such as sodium hydroxide or lime water. That would be if you're investigating aerobic respiration. If you're investigating anaerobic respiration, then you would need to suspend it above a solution that absorbs oxygen. The boiling tube is connected to a manometer with a droplet of coloured fluid in the capillary tube. As the organism respires, it absorbs the oxygen from the boiling tube, causing the coloured fluid to move along the capillary tube. And to measure the volume of oxygen absorbed, you set up one respirometer, but to measure the volume of carbon dioxide, you set up two. Now, if you do want more details on this, I do actually have an entire video on respirometers. Just search on YouTube, Miss Estruch respirometer, and you'll find it just there.
And that takes us to the end of Module 5. I hope you found it helpful. If you did, then make sure that you subscribe so you don't miss out on any of my latest videos.